Pada lantai 3, terdapat ruang dekanat, ruang ketua-ketua program studi S1 dan meeting room. Dan pada lantai 4, terdapat ruang dosen, laboratorium, dan lecture theater. Gedung pendidikan. Pada lantai 1, terdapat ruang pasca sarjana, ruang gugus penjaminan mutu, ruang dosen, dan gudang. Pada lantai 2, terdapat ruang dosen, ruang ketua dan sekretaris senat fakultas, ruang ketua komisi disiplin, ruang kerjasama, ruang senat, ruang sidang, dan fasilitas umum yaitu toilet. Dan pada lantai 3, terdapat ruang-ruang departemen kelautan, yaitu ruang ketua dan sekretaris departemen, ruang dosen, ruang rapat, ruang seminar, dan fasilitas umum yaitu toilet. Gedung penelitian. Pada lantai 1 dan lantai 2, terdapat banyak laboratorium yang digunakan untuk kegiatan penelitian. Di antaranya, laboratorium penangkaran dan rehabilitasi ekosistem dan laboratorium ekologi laut. Pada lantai 2, terdapat ruang kelas pasca sarjana, dan pada lantai 3, terdapat ruang-ruang kelas program studi S1. Gedung laboratorium. Pada lantai 1, terdapat ruang ketua dan sekretaris departemen dan banyak laboratorium. Di antaranya, laboratorium biologi perikanan dan laboratorium kualitas air. Pada lantai 2, terdapat ruang dosen, ruang seminar, dan banyak laboratorium. Di antaranya, laboratorium sistem informasi perikanan tangkap dan geospasial kelautan, dan laboratorium teknologi hasil perikanan. Fasilitas fish hatchery. Fasilitas ini terdiri dari 2 bangunan tidak bertingkat yang digunakan untuk kegiatan pembiakan dan pemeliharaan biota perairan. Fasilitas lainnya yang mendukung kegiatan pendidikan dan penelitian yaitu tambak pendidikan Universitas Hasanuddin di Desa Bojo, Kabupaten Baru, dan Marine Station di Pulau Baranglompo, Kota Makassar. Berikut ini profil infrastruktur umum yang terdapat di Fakultas Ilmu Kelautan dan Perikanan Universitas Hasanuddin. Pelayanan transportasi. Untuk memudahkan aksesibilitas, telah tersedia halte bus tepat di depan fakultas, tersedia pula driveway yaitu jalur kendaraan bermotor dan area parkir kendaraan bermotor yang memadai. Untuk keamanan secara umum dan keamanan parkir kendaraan, terdapat petugas security dan pos penjagaan di depan gerbang masuk. Untuk mendukung transportasi ramah lingkungan, telah dibangun jalur pedestrian di sepanjang jalan utama universitas dan jalur pedestrian yang menghubungkan jalan utama dengan setiap bangunan di dalam fakultas. Terdapat pula jembatan pedestrian yang memudahkan aksesibilitas antar bangunan. Kedepannya, direncanakan sebuah pedestrian boulevard yang akan lebih memudahkan disabel, pejalan kaki, dan pengguna transportasi umum. Selain itu, pengadaan lift, ramp, dan toilet khusus untuk difabel di setiap bangunan. Akan dibangun pula jalur khusus sepeda dan area parkir sepeda yang memadai. Pengelolaan limbah padat. Fakultas menyediakan tempat-tempat sampah di setiap fasilitas dengan minimum 2 jenis pemilahan, yaitu organik dan non-organik. Tersedia pula tempat sampah berskala lebih besar pada fasilitas umum seperti kantin dan laboratorium. Cleaning service setiap hari mengosongkan tempat-tempat sampah yang telah penuh dan membersihkan seluruh bangunan dan area fakultas dari sampah yang berserakan. Pengangkutan sampah dilakukan minimal 2 kali sehari atau sesuai kebutuhan. Konsep Green Waste yang diaplikasikan oleh fakultas secara bertahap mencakup 3R, yaitu reduce, pengurangan sampah utamanya plastik, reuse, memaksimalkan barang penggunaan kembali dan recycle, pengolahan sampah secara mandiri melalui program kemitraan dengan bank sampah lokal dan pengelola sampah kota. Konsep Green Waste juga mencakup pemilahan sampah secara lebih detail, utamanya kertas dan plastik, di mana sampah jenis ini umum diproduksi oleh fasilitas pendidikan. Ketersediaan air bersih. Pelayanan air bersih oleh PDAM telah tersedia 24 jam dengan sangat memadai dan didukung pula oleh sumber bor dan tangki-tangki penampungan untuk menjamin kelangsungan kegiatan fakultas sehari-hari dan utamanya kelangsungan kegiatan pembiakan dan pemeliharaan biota perairan. Infrastruktur air kotor dan limbah. Utilitas air kotor telah tersedia di setiap bangunan dan saluran drainase tersier telah tersedia di sepanjang jaringan jalan lokal yang terhubung dengan baik ke saluran drainase sekunder kampus dan saluran primer kota. Septic tank telah tersedia di setiap bangunan dengan kapasitas yang memadai. Konsep Green Waste juga akan mencakup rainwater harvesting, yaitu pengolahan air hujan dan pengolahan air limbah secara mandiri oleh universitas dan fakultas. Infrastruktur energi dan listrik. Pelayanan listrik oleh PLN telah tersedia 24 jam dengan sangat memadai. Konsep renewable energy yang diaplikasikan oleh fakultas secara bertahap, yaitu berbasis pada solar cell yang memanfaatkan energi matahari melalui panel-panel surya yang ditempatkan di atas atap-atap gedung. Selain infrastruktur tersebut, terdapat jaringan wifi dan hotspot yang dapat diakses 24 jam. Tersedia pula musola dan masjid yang letaknya sangat dekat ke setiap bangunan. Fakultas memiliki taman yang dilengkapi dengan gazebo dan area duduk yang asri. Di antara ragam kegiatan menarik yang dapat Anda ikuti di Fakultas Ilmu Kelautan dan Perikanan Universitas Hasanuddin setiap tahunnya, yaitu pelatihan diving, field trip ke marine station dan pulau-pulau, menjelajah alam bersama organisasi Greenfish, bertanding sepak bola bersama football club, praktek lapang ke Lapan Pare, praktek lapang ke Balai Perikanan Budidaya Air Payau Takalar, seminar nasional kelautan dan perikanan, Marsaf Symposium, International Symposium on Marine Science and Fisheries, dan kegiatan-kegiatan pengabdian masyarakat di desa-desa pantai, di antaranya yang melatih masyarakat untuk keahlian penangkapan ikan ramah lingkungan dan metode pengolahan produk-produk perikanan yang berkualitas. Demikian struktur organisasi, profil bangunan dan fasilitas dan gambaran infrastruktur umum di Fakultas Ilmu Kelautan dan Perikanan Universitas Hasanuddin. Semoga video ini bermanfaat, dan jika suatu saat Anda ingin berkunjung ke fakultas kami, dengan senang hati kami menyambut. Salam hangat dan sampai jumpa.
Your Excellency, the Dean of Faculty of Marine Science and Fisheries, Universitas Hasanuddin, Dr. Insinyur Siti Aisyah Marhum, MSI.
Your Excellency, Director of Institute of Tropical Aquaculture and Fisheries, University Malaysia Terengganu, Profesor Dr. Muhammad Iwanuddin Abdullah.
Your Excellency, Chairman of the Committee of the 8th National and 4th International Symposium on Marine and Fisheries, Universitas Hasanuddin, Profesor Dr. Insinyur Najamuddin, MSc.
Your Excellency, Chairman of the Partner Committee from University Malaysia Terengganu, Associate Professor Dr. Marina Hassan. The Honorable Invited Speaker, Professor James Bell, Victoria University of Wellington, New Zealand.
The Honorable Invited Speaker, Dr. Muhammad Fasrul Hisam Abdul Aziz, of University Malaysia Terengganu.
The Honorable invited Speaker Johnny D. Ong as co-founder, Chief Innovation Officer, Connect AI and Data Science, Melbourne, Australia.
The Honorable invited speaker, Associate Professor Dr. Satoshi Asaoka of Graduate School of Integrated Science for Life, Hiroshima University, Japan.
The Honorable invited speaker Professor Nicholas Paul of University of Sunshine Coast, Australia.
The Honorable invited speaker, Dr. Nurjanda Nurdin, ST.MSc, Head of Center of Marine, Coastal, and Small Island Research and Development, Universitas Hasanuddin.
The Honorable invited speaker Dr. Judy Desways of Rutgers University, Newark, New Jersey, USA.
The Honorable invited speaker, Dr. Yang Yves Mouvel of World Aquaculture Society ASEAN Pacific Chapter.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Let's start our program by reciting Basmalah. Bismillahirrahmanirrahim.
Let us praise to God, the Almighty Allah Subhanahu wa Ta'ala, that today we can participate in the 4th International Symposium on Marine and Fisheries with the theme
The acceleration of marine and fisheries research management to achieve SDGs in COVID-19 pandemic era. Organized by the Faculty of Marine Science and Fisheries, Universitas Hasanuddin, in collaboration with the Institute of Tropical Aquaculture and Fisheries from University Malaysia Terengganu.
Ladies and gentlemen, our first agenda is welcoming remarks as well as opening this symposium officially.
We cordially invite the Dean of Faculty of Marine Science and Fisheries, Universitas Hasanuddin, Dr. Insinyur Siti Aisyah Farum, MSI. To the Dean, time is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, Excellency the Minister of Marine Affairs and Fisheries, Republic of Indonesia, Insinyur Sakti Wahyu Trenggono, MM, representative by Professor Syarif Wijaya, our Honorable UNHAS Rectors, Professor Dwi Aristina Pulubuhu, and all Vice Rectors, Director of Institute of Tropical Aquaculture and Fisheries, University Malaysia Terengganu, Professor Dr. Muhammad Ihwanuddin Abdullah, Chairman of the Committee from Universitas Hasanuddin, Professor Najamuddin, Chairman of the Partner Committee from UMT, Associate Professor Dr. Marina Hassan, my colleagues, deans, vice deans, and leaders of marine and fisheries related faculty, department, and study programs from all over Indonesia. Distinguished speakers, Professor James Bell from Victoria University of Wellington, New Zealand, Dr. Muhammad Fazrul Hisam Abdul Aziz from University Malaysia Terengganu, Johnny Ong, co-founder, Chief Innovation Officer, Connect AI and Data Science, Melbourne, Australia, Associate Professor Dr. Satoshi Asaoka from Graduate School of Integrated Science for Life, Hiroshima University, Japan, Professor Nicholas Paul from University of the Sunshine Coast, Australia, Dr. Nurjana Nurdin, STMSI, Head of Center for Marine, Coastal, and Small Islands Research and Development, Universitas Hasanuddin. Dr. Judith S. Weiss from Rutgers University, Newark, NJ, USA, Dr. Yang Eve Mewel from World Aquaculture Society, Asian Pacific Chapter. Respected guests and participants to the 4th International Symposium of Marine and Fisheries Issue in Makassar. First of all, as part of our religious and cultural tradition, I would like to invite everyone to pray to our Almighty God to always bless us and protect us all, especially in this pandemic situation. May of us always in good health. As the Dean of Marine Science and Fisheries, Faculty of Universitas Hasanuddin, who hosts this symposium, would like to thank all parties, organizing committee, UNHAS leaders, government, speakers, who have been helping and collaborating with us significantly, without whom the symposium will have been impossible. I would like to especially thank to Ibu Rector, who always encourage and give support to conduct this international symposium as part of national and global commitment to participate in science and technology development to understand the problems and provide solutions. Also, thanks very much to all speakers today, who have been very kind and enthusiastic to participate in this online symposium. As I said last year, that we hope in the year 2021 we can meet again face to face in Makassar, but unfortunately this pandemic prolongs and we can only wish and pray that we can get through this difficult moment, and may all of us safe until the normal day. We might call it the new normal day will eventually come sooner rather than later. To all participants and presenters, thanks a lot for your contribution to make this virtual symposium finally taken place today. Again, we still remember 2 years ago when we conducted the second international symposium on marine and fisheries issues here in Makassar. At that time, we all gathered in the hotel, meeting each other face to face, shaking hands, drinking and eating together while enjoying jokes, conversations, and so on. Although we cannot do the same way as we did it in the past, I do hope that every one of us keep the spirit of sharing, collaborating, and helping each other to pursue knowledge and skills to solve the common challenges related to marine and fisheries issues that we all facing now and in the future, not only here in Indonesia, but in all our countries and around the globe. Distinguished guests, ladies and gentlemen, our symposium this time choose specific theme: acceleration of marine and fisheries resource management to achieve sustainable development goals in COVID-19 pandemic era. We need innovation, improve resource management to achieve SDG targets. At the same time, we need adaptive strategies to adapt with uncertainty. For example, as we are now experiencing during this pandemic COVID-19. Innovation and technology have a very important role, especially in increasing the competitiveness of Indonesian fisheries products at the global level. Adaptive strategies for aquatic resource utilization and management also urgently needed to improve quality and quantity of production and welfare without harming the sustainability of environment. All of those require science, knowledge, skill, technology, strategic management, and policies that we need to share and develop together. We expect the encounter of many people from many institutions in this virtual symposium, not only resulting new concepts, but also building and strengthening the cooperation of various parties by all of us. I also hope through international collaboration, we can contribute to improve the quality of global marine affairs and fisheries sectors to meet the interest and expectation, not only scientists, but also industries, private sectors, marine and fisheries households, NGO, as well as governments. So let's get to work smartly, collaboratively, and openly as friends, colleagues can and should. I look forward to seeing the presentation and sharing process in this symposium. Let's continue strengthening of this bond of friends and colleagues who are among the participants today despite these pandemic challenges. With this, I conclude my remarks and wishing you all the best. Stay healthy, happy, and productive. I'm looking forward to personally seeing you all again in the near future. Thank you. Good morning and wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Dr. Insinyur Siti Aisyah Farhum, for your delightful welcoming speech.
Dear ladies and gentlemen, to maintain safety and comfort during the symposium, all of the participants are required to obey this code of conduct.
First one, presenters or participants should dress neatly during the event and use the virtual background provided. Second one, plenary session, active video and sound are only for the keynote speakers and the moderators. All others attendees should mute their Zoom audio.
Ladies and gentlemen, our next agenda is the main agenda of the third International Symposium on Marine and Fisheries, the plenary session.
For the plenary session, we will have 8 excellent main speakers. This session will be led by Ms. Asmi Chitra Malina, SPI, Master of Agriculture, PhD, will ask the moderator. Please welcome Ms. Asmi Chitra Malina.
Thank you, Adnan and Suci. The Honorable Dean of Faculty of Marine Science and Fisheries, Universitas Hasanuddin, Dr. Insinyur Siti Aisyah Farhum, MSc. Director of Institute of Tropical Aquaculture and Fisheries, University Malaysia Terengganu, Professor Dr. Muhammad Ihwanuddin Abdullah. Chairman of the Committee of the 8th National and 4th International Symposium on Marine and Fisheries, Professor Dr. Insinyur Najamuddin, MSc. Chairman of the Partner Committee from University of Malaysia Terengganu, Associated Prof. Dr. Marina Hassan. The honorable invited speakers, distinguished guests, all the participants, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. May peace be upon us. Good morning to those in Indonesia, good afternoon and good evening in the citizen of the world. First of all, I would like to welcome you all to the 8th National and 4th International Symposium on Marine and Fisheries, Faculty of Marine Science and Fisheries, Universitas Hasanuddin, in collaboration with University Malaysia Terengganu. It is a wonderful conference with two sessions from this morning to this afternoon. It is a great joy and privilege to me moderating the symposium with the theme The Acceleration of Marine and Fisheries Resource Management to Achieve Sustainable Development Goals in COVID-19 pandemic era, with the all prominent speakers that we have today. Ladies and gentlemen, before we start the first sessions, I'd like to say hello to the invited speakers today. We have 8 speakers. The first one is Professor James Bell from Victoria University of Wellington, New Zealand. Hi, Professor James Bell. Are you there?
How you doing?
Good morning. Thank you. And the second one, Dr. Muhammad Fazrul Hisam Abdul Aziz from University Malaysia Terengganu. You there?
Hi, I'm here.
Oh,
Yeah,
okay.
can you hear me?
Yeah, I can hear you clear. Good
Okay,
morning.
okay, thank you.
And the third, he's my friend from Melbourne. He's my classmate in high school, Mr. Johnny Deong, co-founder, Chief Innovation Officer, Cogner AI, Artificial Intelligence, and Data Science, Melbourne, Australia. Please correct your company name if I made mistake.
Yes. Good morning,
Selamat
Asmi.
pagi,
That's
Bapak
correct. That's
Johnny D.
Cogner. Yeah, AI data
Masih
science
ingat
in
bahasa
Melbourne.
Indonesia, Pak? Do you still remember bahasa?
Of course.
Okay. Yes. And the fourth is associated from Professor Dr. Satoshi Asaoka from Graduate School of Integrated Science for Life, Hiroshima University, Japan. Ohayo gozaimasu.
おはようございます。
お元気ですか?
Thank you very much. I'm fine.
Okay, good. And the fourth is Professor Nicholas Powell from University of the Sunshine Coast, Australia. Nick, are you there? Oh, maybe he will join later. Yeah. Okay, the next speaker, Dr. Nurjana Nudin, STMSE, Head of Center of Marine, Coastal and Small Islands Research and Development, Universitas Hasanuddin. Assalamualaikum. Good morning, Bapak Ibu Nurjana.
Assalamualaikum
And
warahmatullahi wabarakatuh. My sister
next
Dr.
speaker
Rasmi.
is Dr. Judith Esways, Rutgers University, Newark, New Jersey, USA. Judith, are you there?
Hello,
Hi.
can you hear me?
Yeah, I can hear a long time.
It's been a long time. It's very good to see you again,
Yeah,
Sitra.
very good to see you too.
Good to be
Thank you.
here.
Yeah. The last you visited in Bilibili, yeah? Goa. Okay. And the next, last but not least, Dr. Jane Yves Miller, World Aquaculture Society Asian Pacific Chapter. Good morning. Are
Yes.
you there?
Good morning. I'm not sure if you can see me, but I can see all of you.
Okay.
I don't know why it's not working for me. The camera, it looks like.
Oh.
Is
Okay.
it?
It's okay. Yes. Thank you very much. Ladies and gentlemen, we now we started to the first sessions. So I would like to invite the first speakers, Professor James Bell from Victoria University of Wellington, New Zealand. Please allow me to read his biography. Could you please share? Professor James Bell, a marine biologist in the School of Biological Sciences at Victoria University of Wellington, and is currently the Marine Biology Program Director. Graduated from the University of Wales, Bangor in 1998 with a first-class honors degree in marine biology and gained his PhD in 2001 from University College Cork in Ireland. He became intrigued by sponges during a research expedition to Lough High Marine Nature Reserve in Southern Ireland. And his PhD research focused on the unusual spongy assemblages there. Professor James moved to New Zealand to take up a lectureship at Victoria University in 2006 and was promoted to professor in 2018. He is an internationally recognized marine ecologist whose current research is at the leading edge of spongy ecological research, particularly focusing on temperate and tropical ecosystem. Professor James Bell work of interested is the understanding the functional roles of sponges in marine system, the ways in which human impacts influence these roles, and the subsequent ecosystem consequences of declines or increases in spongy abundance. So Professor James Bell has published over 140 peer-reviewed journal articles and book chapters in leading international journals, including a number of recent high-impact articles. We welcome Professor James Bell to give presentation. Your time is 20 minutes from now. Please welcome.
Great, thank you very much. Can you hear me and see me? See my slides?
Yes.
Yes, great. I guess I'd just like to start by thanking you very much for that kind introduction. And thanks everybody for I guess tuning in today to listen to me, and thanks to the organising committee to putting this together. I was actually at the last conference, I believe, and I'm really hoping at the next one I'll also be at that one and can actually see everybody in person again. Obviously a very, very difficult time for everybody at the moment. But I'm really delighted to be here this morning to get the opportunity to tell you a little bit about my research. You might think that, my research is focused on perhaps a slightly obscure group of animals. All of my research in Indonesia and tropical reefs across the world is focused on sponges. And hopefully what I'm going to tell you about in the next 20 minutes or so is just a little bit about why I think you should care about sponges and about some of the research that we've been doing over the last kind of 10 or 15 years, which includes quite a bit within Indonesia and the wider Indo-Pacific region. I guess the first place to start is to try and convince you why sponges are actually important. So sponges are found in temperate, tropical and polar ecosystems across the world. And one of the primary things that they're known for doing is pumping large volumes of water. So what you're looking at here is a small sponge about the size of half of your fist. And we've labeled the water surrounding the sponge with a dye called fluorescein. And you're looking at this sponge pumping the water. So sponges are pretty simple organisms. They are a group of, they're obviously multicellular animals with a few different cell types with a body plan that's specialized to pump water. That's effectively what a sponge does. It has some specialized cells called choanocytes that beat backwards and forwards and generate this water flow. As they suck in this water, they remove the particles that are in the water column with very high efficiency. So our research groups and others across the world have found that they basically feed on things like bacteria and phytoplankton, and they remove those small particles at extremely high efficiency. So they have a really important role in moving material, particularly carbon, from the water column to the seafloor. That used to be the kind of paradigm that people thought happened, is sponges were mostly responsible for moving carbon down onto the seafloor, and it kind of got locked up into the sponge. What you're looking at here is a time-lapse photography over about 3 hours of an Indonesian sponge called Spongia. So this is obviously speeded up. Hopefully what you notice is that there are those three worm-like things crawling over the surface. They're actually a type of sea cucumber that are basically crawling over the surface of the sponge and feeding on mucus that the sponge is producing. Hopefully what you can also see is lots of little fish and tiny stuff turn up. They're taking little nibbles of the animals that are associated with the surface of the sponge, also feeding on the mucus that's being produced to try and get rid of the sediment that's on the sponge. And also what they're feeding on is detritus that the sponge itself is producing. Those cells that create the water current through the sponge turn over very quickly and get exuded by the sponge and eaten by other stuff. So we now know that sponges not only remove lots of stuff from the water column, but then they also recycle that material to other parts of the ecosystem. Sponges also have another range of important functional roles on tropical reefs. Firstly, they provide habitat for the species. If you chop open a sponge, you'll find that there's lots of different stuff living inside them. And that's really important because all of those things end up as food for other stuff. They're also really important as eroders of substrate. So while things like corals are making the calcareous structure on a coral reef, sponges are one of the primary agents that are breaking down that calcium carbonate. So if we lose corals and we get an increase in these bioeroding sponges, then you can see a situation where the overall reef structure will eventually break down. Sponges are also an important food source for lots of different things, particularly for turtles, for fish, and for nudibranchs, for sea slugs. And there are many of these that are quite specialized feeders. Also, really importantly, is their role in what we would call facilitating primary production. So sponges form a number of relationships with lots of different symbiotic microbial organisms, and in particular, they form a lot of relationships in the Indo-Pacific with cyanobacteria, so photosynthetic organisms. And some of our recent research has demonstrated that not only do these cyanobacteria produce enough carbon to feed the sponge, but also during light periods, these sponges are then releasing carbon, photosynthetically derived carbon, into the surrounding water column. So sponges are at the center of all these different trophic interactions and these carbon flow pathways. Of course, these functional roles are only important if sponges are abundant in ecosystems. And what you're looking at here is some work we did a couple of years ago, the little mini ROV that we have here at Vic, looking at changes in the coral and sponge abundance with depth at one of our kind of main study sites. And what you can see is a video here is about 60 or 70 meters. And all of those big kind of large structures are all sponges. And as you go deeper down on most coral reefs, you'll find a transition at about 20, 30 meters where the coral starts to decline because there's not enough light for their symbionts. And we find these massive increases in sponge abundance. In most places in the world that I've worked, and Indonesia is not really any different, when you're around kind of, 10, 15 meters or shallower, sponges typically on a healthy-ish reef will occupy about 8 to 10% of the available substrate. But once you go down below 50 or 75 meters, you find that sponges are probably the most dominant organism, the most abundant organism. So in many respects, coral reefs already are dominated by sponges. And we need to know about these ecosystems because these ecosystems are supporting a lot of stuff that's going on in the shallows. So what I want to focus kind of the last bit of my talk around is really a central hypothesis that drives a lot of my research, or has done over the last kind of 10 years or so. And that's centered around an idea that I came up with a number of colleagues of mine, that many of our coral reefs, as they're declining, may actually transition to other different states. And those states, or one of those states, could be a more spongy type environment. And I want to talk about some of the evidence to support that hypothesis. Now, we all kind of know the threats that coral reef environments face. These are graphs that I'm sure you will have seen before, and kind of messaging that is common across all media. Coral reefs under decline. The Indo-Pacific generally isn't so badly hit as perhaps areas of the Caribbean, where coral cover is exceptionally low in most places. In fact, in the Caribbean, there are a few exceptions to that, but coral cover is generally very low. And the common paradigm in the literature has been that when the corals decline and disappear, then macroalgae comes in and takes over, that space that was once occupied by coral. However, in not all cases does that happen, and there are lots of examples, particularly across the Indo-Pacific, where reefs where the coral has disappeared are not taken over by macroalgae. In some cases, nothing appears. In some cases, you get turfing algae. Some cases, you get things like ascidians, and in many other cases, you get an increased dominance of sponges. So I want to talk about my work in kind of three contexts. The first of these, I want to describe a shift in a coral reef that we've been working at for quite some time, where corals were once dominant and then have taken over. And then I want to talk about some of the resilience work that we've been doing on sponges to kind of demonstrate to you how resilient we think they are to lots of environmental impacts that are causing declines to coral. So kind of central to this hypothesis is the idea that if sponge reefs are going to be something that we're going to see more of in the future, that means the sponges must need to be more tolerant to the things that are causing declines in our corals. So this is Wakatobi Marine National Park. I'm sure many of you in the audience are familiar with this site, and hopefully I'm sure many of you have visited it as well. Obviously, it lies at the centre of the coral triangle, highly biodiverse place. But like all coral reefs, is under threat from basically a whole range of human impacts. I guess there are somewhere in the region, maybe 100,000 people who live in the park who are directly reliant on the reefs around this area for food, resources and building materials, and an area where lots of waste goes into the sea as well. So I've been lucky enough to be working here since about 2002, 2003. And here's some data for 6 different sites in the Wakatobi between 2002 and 2007. And this was actually data collected by a PhD student in University of Essex, not from my group. And hopefully what you can see is sometime around 2004, 2005, there was a pretty large decrease in coral cover in most of these sites. We actually went back in 2016 and did some further surveys with people from my group. And we found that this decline has continued, sadly, and you can see in some of these sites the declines have been really, really dramatic. For example, over here at Caldupa, one of our sites, you can see coral cover was at 40 to 50%, and now it's around about 10%. I want to particularly focus, though, on this site over here called Sampella. Its coral declined very rapidly from about 30% down to about kind of 5 or 6% now. And this site is characterized by having very high turbidity and very high levels of sedimentation, primarily or most likely as a result of a Bajau village that is very in close proximity to this area. It's a very highly degraded, highly sedimented site. The thing about this site is it's very much dominated by sponges. We've been studying the sponges at this site for over 15 years now. And one of the species we've been particularly focusing on is this one here, which is Lamella Dicidia herbacea. And what you're looking at here is the abundance of this species. This was in 2015. At 4 different sites in the Wakatobi, including Sampella. And you can see at Sampella, there is a really high abundance of this, about 20% of the available substrate, which declines with depth. The reason its abundance declines with depth is because this is one of these photosynthetic species. It has cyanobacteria producing carbon for it, and we know it's really reliant on that. If we compare that data with data that I collected back in 2003 for Sampella, unfortunately, I didn't sample the other sites back in 2003. I wished I had, but never did it. You can see that there has been a large increase in the abundance of this sponge at all the depth. So about 10% of the area was covered in the shallows back in 2003, and now it's up to about 25%. My PhD student who did this work also monitored changes in this species over time between 2013 and 2015. Again, these are data for Sampella, which are the triangles here, the squares, and then the circles. And then for another site, Ahoga, basically a more pristine or a better quality reef. And hopefully what you notice on the reef flat is the abundance of this species still seems to be increasing. It's taking up all the available space. The other thing about this Sampella system is it's not just this species that's super abundant. We don't have long-term data for the other sponge species, but at this place, this is the place where barrel sponges, for example, those big, giant monster sponges, those are also the most abundant at this site. And we also see a whole range of other different shapes and morphologies that are just kind of carpeting the seafloor and have taken over that space from the corals that have declined. So that's one example. I've actually worked at probably 10 or 15 different locations now across the globe where we've seen these declines in coral cover and we see these subsequent shifts into sponge assemblages. And I think as more people look for these things, they're finding more of these types of environments. The next thing I want to talk about is the ability for sponges to withstand the impacts or the things that cause declines to coral, because that's kind of critical in determining whether or not sponges are going to be a more dominant feature of the organisms that we find on reefs in the future. Now, our group has done a lot of work with sedimentation. So we know from the literature that sedimentation is generally detrimental to corals. Not all corals, but it's generally detrimental to most corals. And that's because they've got their photosymbionts as well. And their symbionts need light, and sediment often shades out that light or settles on their surface and then stops the light getting to the symbionts. So this is, again, another time-lapse photography you're looking at from the inside of a giant barrel sponge. And this was work done at Sampella. And what we found is that if we stick sediment on these sponges, so if sediment falls on them from above and settles on them, the sponge produces large amounts of mucus. And that's what you're looking at in this picture. This is obviously sped up. The sponge produces loads of mucus. The mucus binds all around the sediment. And then that basically, as more mucus is produced, the density of it decreases. And the exhalant water current of the sponge carries away the mucus and sediment. It's a really amazing mechanism for ridding itself of sediment. We've also done work on this species Lamella Dicidia herbacea to see how that tolerates the sediment. Again, you would think for this species, because it's also reliant on photosynbionts, that living in a sedimented environment might not be the best place for it. But it too produces large amounts of mucus, which basically bind together with all the sediment and then slough off the top of the sponge. So it has these mechanisms to make sure that light still gets to its symbiont and it gets all the sediment off its surface. This graph here shows we did some experiments where we put some sediment on top of these sponges, and we saw how long it then took for the sediment to be taken away from the sponges. And basically after 4 days, if you put sediment on these sponges, quite a lot on, all the sediment is gone by these mucus trails and this mucus production within 4 days. And they're really good at doing that. The problem for the sponge is it's an energetically costly process. So we found that when we basically dump the sediment on the sponges, first of all, the sponge stops using energy, it stops pumping. That's probably because it goes into some kind of shock. Then as it starts to initiate this mucus production to get rid of the sediment, it uses quite a lot of energy to do that. And that's important from an energetic perspective, because if it's spending energy on removing sediment from its surfaces, it means it doesn't have as much energy for things like growth and for reproduction. The other thing that we've worked on recently quite a lot is the impacts of nutrient stress on sponges. So obviously we all know that there are increased nutrients from things like agriculture and things like sewage, and the Wakatobi and other areas in the Indo-Pacific are no exceptions to these impacts. And what we found, perhaps a little bit surprisingly, is that it doesn't seem to matter what we expose sponges to nutrient-wise, they're really tolerant to this. And again, this is different to corals. Although there's some mixed responses seen between corals, generally across the phyla, increases in nutrients are thought to be detrimental to most coral species. As I say, there are some exceptions to that, and it does depend on a kind of location by location, study by study basis. But generally, there's quite a lot of negative impacts of increased nutrient stress on corals. Whereas for sponges, they just don't care at all. So at least for these two stressors, and most of the other ones that we've looked at, sponges appear to be a bit more tolerant than what corals are. Final thing I just want to talk about is the resilience of sponges to large-scale environmental impacts, and particularly in this case, climate change. If sponges are going to be a more dominant feature on reefs in the future, then they're going to need to deal with the impacts of climate change better. We've done a number of experiments over the years looking at the impacts of climate change on sponges. We have some experiments that finished a few years ago in collaboration with the Australian Institute of Marine Sciences in Townsville, where we used their really cool sea simulator to expose sponges to present-day conditions and conditions under 2 of the IPCC climate change scenarios. And we worked on a number of common species that you find across the Indo-Pacific. I just want to talk about the results of one of those species. There are, as you can imagine, it was a 3-year project and generated a number of publications. But I just want to talk about one of the most interesting things, or at least that I think that we found. What you're looking at here is information for one common Indo-Pacific sponge. This is Carteria spongia foliaceans, found across the Indo-Pacific and including Indonesia. And what you're looking at as a response variable here is necrosis, which is basically how much of the tissue is gone all manky and nasty and breaking down, and how much of the sponge is bleached. Now, this sponge is a photosynthetic sponge. It has these cyanobacteria in its tissues. So if it's bleached, then it loses color. So these are the results from the 28.5 degrees, so normal conditions. This is the middle IPCC scenario, and this is the worst case scenario. And as you can see, hopefully here, so this is the normal conditions, normal temperature, sorry. If we reduce the pH, so we experience ocean acidification, the sponges aren't impacted at all. There's no bleaching or necrosis. At the 30-degree treatment, so middle IPCC scenario, once we get down to the lower pH, we do start to see some impact on the sponge, but again, quite small. Perhaps the most interesting result, though, is when we expose them to really high temperature. If the pH stays the same, then basically all the sponges die and bleach. But if you reduce the pH at the same time, then we find that sponges are better able to tolerate the heat. So if they experience ocean acidification and they experience high temperature at the same time, we expect some of these photostrophic sponges most likely because of their relationship with the symbiont to be able to survive these conditions. In contrast, if you exposed corals to these conditions, pretty much all of them die. The final thing I just want to mention, I think my 20 minutes is nearly up, is I just want to talk a bit about how a sponge reef might function. You might think, sponges are a bit obscure group of animals to work on, but you may find that over time, many reefs transition to states that are more dominated by these animals. And this is going to have real big impacts for how reefs actually function and work and the resources that we can take from them. One of the things my group has been working on is how having a sponge-dominated reef can influence overall reef complexity and habitat complexity, because we know that's really important in productivity, and we know that's really important in supporting biodiversity. And we use some quite cool 3D photogrammetry to study and to explore how this might change that habitat complexity and the spaces available on reefs for organisms to actually live in. And what we found is with sponge-dominated reefs, there were lots of bigger holes still. So larger fish had somewhere to live and hide from their predators. But we found there was a big reduction in the number of small spaces. And of course, those are really important because that's where all the small stuff that forms the basis of the food chain lives and where all the juvenile fish hang out so they don't get eaten by predators. So although you're probably still going to have lots of fish on a sponge-dominated reef, the overall productivity is going to be lower, much lower compared to a coral-dominated one. And just really my final data slide, we've also been doing some stable isotope analysis to look at the impacts of a sponge-dominated reef on overall food chain. What you're looking at here is a stable isotope.
5 minutes left, your time.
How many?
Sorry, 5 minutes.
Cool. Oh, good. I'll speak slow. Good. So one of the one of the things that my group's been looking at as well is how food chains are influenced by being a sponge-dominated or a coral-dominated reef. And what you're looking at here is a stable isotope data for a sponge reef at the bottom and a more high-quality reef at the top. Now, each one of the colors represents the stable isotope data from a different fish species. Now, there are a whole range of different stable isotopes that we can use, but typically we use carbon-13 to give an indication of what things are feeding on. And we use nitrogen-15 to determine where in the trophic scale, food chain, an organism sits. So each one of those dots represents the data from an individual fish. And hopefully what you can see is for most of these fish species, on the sponge-dominated reef, the data is much more widely spread than it is on the coral-dominated reef. And the reason for this is that these species that are living on the sponge-dominated reef end up having a smaller range of food sources. There's a lower range of carbon sources for them to feed on. And also they have to search out more types of food. So their dots and their stable isotope signatures are more widespread because they're having to feed on a much wider range of things. And that's even for fish species that are known to be obligate spongivores and obligate corallivores. Within these environments, they're actually having to switch and feed, in these sponge-dominated environments, having to switch and feed on different stuff, which again is going to improve productivity. And so just in summary, hopefully I convinced you a little bit at the beginning that sponges have important functional roles in marine ecosystems. And my research, not just from the Indo-Pacific, but from coral reefs across the world, are suggesting that sponges are more tolerant to the impacts of local scale degradation, things like sedimentation and nutrients, and also large scale impacts like climate change than what corals are. And what we're seeing as a result of this, because one of the controlling factors of sponges on reefs is that spatial competition with corals. And when those corals decline, those sponges are taking up that available space. Although we're really only at the start of understanding what that will mean from a kind of overall ecosystem perspective, we do expect it to have really profound impacts on the overall food chain and ecosystem function, particularly the overall productivity of a reef. I don't want to give you the impression that sponge reefs will be, completely devoid of fish and resources. They will provide food resources, but they're not going to provide the same level of resource as a healthy coral-dominated reef. And it really highlights why we need to make sure we conserve coral reefs and we remove all these stressors so that we don't get these novel and unusual ecosystems. I guess like to acknowledge there's a whole lot of people who have contributed in various ways to this project, a whole lot of different people who have provided funding. And I thank you very much for your time, and I hope you have a good day.
Thank you very much, Professor James Mill. And I think you got the time, yeah? 20 minutes. And we move to the next speakers. We would like to invite Dr. Muhammad Fazrul Hisam Abdul Aziz. Let me read your profile, short biography. Please share.
Okay.
Activity.
Assalamualaikum and very good morning, everyone. Okay. Thank you to the moderator. Okay, right now I would like to present my presentation. The title is about the overfishing pressure and impact of bycatch to the fish wild stocks. So, first of all, for the fishery status, we have around 1,500 fish stocks around the world. Okay, so just 500 stocks are recovered. Okay, and for according to Sophia report, there are depleted stocks has increased from 30% in 1974 to 39.9% in 2020. Okay, the total annual fish has peaked in 1994 at 63.3 million tons. In 2018 is to 19.2 million tons was landed. It's about 5 times more than in 1950,
Dr.
12.8 million tons.
Excuse me, could you please make a slideshow?
Hello?
Slideshow, please.
Slideshow, sorry.
Yes.
I'm sharing already, sorry.
No. Yeah, no.
Can you see it?
Is that slideshow?
Or not? No. Sorry.
Okay.
Can you see the slideshow or not?
Oh, okay. All right.
Okay, can you see it? Can you see it?
Yes.
Yes.
Yeah.
All right. Okay, get it. Okay. Okay. Actually, I'm working on my field is fishing gears and fish ecology. So I'm focusing now right to determine the bycatch from artisanal fishermen. So my publication first is on crab gill net. Actually, the crab gill net is very famous in Malaysia and Asian region actually. And more than 20 kilometers around UMT, our university, they use the gill net around. The length of the net is 2 kilometers. Can you imagine the depth of the net is around 4 meters. So the impact of the bycatch is very high. Okay, so the category of bycatch, we can divide it into 3 categories. So we call first normal. Normal fish, normal bycatch is you try to catch the target species and you get the other species, we call it normal fish, normal bycatch. For the ghost fishing, actually your fishing gear is lost in the sea during the fish operations, but the fishing gear still can catch the fish and still active. So we call it ghost fishing. And for the cryptic bycatch is if the fish entangled into the net and they capable to release themselves, but they get injured and die. We call that cryptic bycatch. We have 3 categories. This is example of the bycatch that we get from the artisanal fishermen. That they just use crab gill net, pelagic gill net. In the left side, you can see here is a bull shark. And this kind of race, that both of these kinds of species is not matured yet. So it's quite big and it's quite sad to see this kind of activities happening almost every day. And also the bycatch definition can be determined by the local perception. For example, this kind of shell in Malaysia, in the up one, we didn't eat this. So most of this bycatch will be living dead on the land. Okay, and at the lower one, we can see this kind of centipede prawn. We also rarely eat this, and this kind of shark we just take the what we call the fin of the sharks. It almost happen every day, and from the artisanal fishermen around our campus, we can get around 200 to 300 kilograms bycatch daily. Okay, this kind of bycatch is very expensive. We call it golden sandfish. Most of the fishermen try to dry it, and we can get around $100 for each kilogram for the dried one. So this is incidental bycatch, and we catch it seasonally. Okay, how bycatch can contribute to fish stock decline? As you can see here, well, from my previous study, and I published already, 40% of the crab is buried female. So we catch a lot of buried female entangled to the crab gill net. So what I try to educate the fishermen is try to educate them to release the buried female or hatch the crab at their home. So we have three to four stage of crab eggs. The last one is black. As you can see, we also have lobsters, and we also have squid. If you pull out the net from into your boat, sometimes you can see a lot of squid eggs. Okay, this is by catch with high value. So as you can see here, we have lobsters, we have golden sandfish, we have slipper lobsters. Okay. Okay, this is invaluable bycatch. As I mentioned before, most of the fish didn't reach the market size, so the market size is very what we call very cheap, and they don't have demand on the market. So most of these bycatch are live dead on the land. So what I can tell you, this is happen almost every day by the artisanal fishermen, including the modern fishing gear. For example, bottom throw net. Okay, this is from the gill net that around UMT. As you can see, this is just one gill net. Can you imagine one fisherman actually use 10 gill nets? This is just from one gill net. The impact is very high. Okay, how to reduce bycatch in fisheries? So what I'm doing, we have two level of sensing the fish in the open water. First, we call it macro level. We use oceanographic satellite, and the other one is airborne, and the last one is radar. But the accurate one, we can use the micro level one. We use echo sounder or sonar, or we call it acoustic instrument. So for scientific purpose, we can use scientific echo sounder that can collect target strength. Okay, this is the publication, and I have been working before. We estimate the pelagic stock in the peninsula of Malaysia. Okay, so this is the basic concept to detect the fish, but we still depend on the swim bladder of the fish. Okay, because we want to detect the pelagic fish and also the behavior of the fish. So as you can see here, the brown color have the strongest signal that indicate there are fish underwater. Okay, before we every type of electronic instrument have calibration, so we have to calibrate the transducer we call underwater. Okay, this is kind of the instrument that we use for our study. We call this Furuno. It's from Japan. So the software also from there. This is the result before. We have 17 echogram that indicate of some Decaturus marwatsi, 11 echogram of Megalopsis cordilla and Celabux. We combine our acoustic instrument with the Midtronet for the scientific purpose. Okay. So we also have threshold analysis. Threshold analysis is we want to remove the noise. We just want to take the what we call the fish target only, the data of the fish only. So we have to remove the noise of the echoes. Okay, this is how we set up in the boat. Yeah, on the vessel we have computer, so we just put this amount. We have to calculate it first, but we calculate it manually and we put the value in on the vessel. So this is SSV upper threshold, and this is the equation that we use: near surface zone, bottom depth zone, fish aggregation, and ping rate. Okay, we just put the value inside the software on the vessel. So how we want to calculate a group of fish and or single fish, but the basic one, we have to know the target strength first before we can estimate fish stock in a massive area. So this is the equation that we use. For example, this target strength. This is the single fish. For the density, we call it SV or SA. This kind of instrument must be get from the software. Okay, this is how the vessel move. 0.1 mile per ping. Okay, one ping, the sound of speed underwater is 1,500 meter per second. Okay, before we go to the wild stock, we have to control our experiment first. We put the species in the cage underwater. So each single fish can produce one single ping that give a unique target strength. That target strength, we make some equation, linear regression, that we can y equal to mx plus c, that we can use it to estimate the wild stock in the open sea. This is the echogram that we get from our control experiment. As you can see here, it is just single dot, but we can go for more details. We can get this kind of data. For this kind of target strength, we get minus 40.12. The target strength will indicate the long, the weight, and something on. Okay. Okay, I go for this is from the wild stock. Okay, I want to show you the important part. For example, from this kind, this is the area that we estimate the fish stock is very. This is peninsular Malaysia. Can you imagine? We have 3 big area, and this is the movement of the vessel. We have to go for zigzag. We cannot go randomly. We have to go for zigzag to estimate this kind of area. We have area A, B, C in the East Coast Peninsular Malaysia. And we, based on our scientific echo sounder, we can determine which depth that the pelagic species is what we call assemble. For example, in this study, we find that the pelagic species only focus on 15 to 20 meters only. Okay, they did not go further for 40 to 60 meters. So the fishermen can focus on the 20-30 meters only to catch the matched fish. Okay, if they go for underwater and more deeper, they will get smaller fish. Okay. Okay, this is a kind of work that we use, that we make at UMT to reduce bycatch. First, we know that fish attracting to light. So in Malaysia, fish behavior is very unique. It's different between habitat and seasons. So in Malaysia, most of the pelagic fish, squid, attract to the light, green light. So this kind of work also has been published just recently, okay? And for the fish gill net, like I mentioned to you before, this kind of work is with the artisanal fishermen around our campus. So before this, they use some kind of net that just entangle almost everything. So what I'm used to work with them, we try to design a new net that can entangle only matched fishes. Okay, so this kind of fish that we get the matched fish, no small fish. We can reduce around 90% bycatch. Okay, and this is also the squid net. The squid net is the first design in Malaysia. Before this, we didn't have squid net. So the squid net, the squid stock in Malaysia is quite stable. So what I'm doing before this, we just go for squid jigging. But for this, I'm telling them we try to design the squid net so we can get a lot of squid with what we call with a less time. Okay. So this is crab gill net. Before this, the crab gill net contribute 52.2% of bycatch, but we try modify back the crab gill net, and right now the crab gill net just can reduce 95% of bycatch. So it's quite good for their economics, and we can reduce bycatch. Okay, this is the lobster and crab trap that we design because at UMT we have a big breakwater. The breakwater is built by a big rock. So most of the lobster try to make this kind of rock for their habitat. So I designed the lobster and crab trap. This is the result. So as you can see here, we can get a lot of crab and trap. Crab and lobster. Okay. For the fish trap, it's actually common worldwide, but at Malaysia, we just use one size. I try to educate the fishermen by combining two mesh sizes all together, so the smaller fish can go out. And we also redesign back the entrance part. So most of the fish, we can get a bigger fish and a lot of what we call very expensive fish like lobsters, and this is kind of demersal fish and grouper. Okay, this is also the squid trap. Okay, the squid trap, before this we didn't use coconut leaf in Malaysia, but based on my study, I also have published it last year. The squid is attract with the coconut leaf because they found it the coconut leaf can give a strange smell or odor to attract the squid. So most of the squid will come inside the trap with the coconut leaf is there. So I tried to design this, and right now most of the fishermen around our campus use this kind of trap already. Okay, this is the squid that we get from the trap. It's quite big and matured already. Okay, this is the prawn net that we use around UMT. Actually, around our campus, the prawn distribute seasonally. It's peak around June to August. So I tell them to try to make this kind of net that just catch only prawn only. So it's quite expensive. In Malaysia, it's around $30 per kilogram. Okay. So for the sustainable fisheries, like I mentioned to you before, we catch a lot of buried female.
Sorry, Dr.
We try to educate the fishermen by take the buried female and hatch the buried female at their homes. So they just put in some kind of tank, put some aeration for 2 days, and the crab will hatch. The eggs of crab will hatch, and then we release it in the open sea.
You have 5 minutes left. Please.
So we also have the buried. Okay, so do the same things. Okay, this is for example, this is some kind of squid that we get that entangle to the net. So we take the squid eggs and we try to hatch it in the tank. So this kind of work we do almost every day. So if they get the squid eggs, the crab eggs, we try to release it in the open sea. Okay, thank you. That's all from me.
Thank you.
Okay, right.
Yeah, thank you very much to Dr. Muhammad Fazrul Hizam. Before we move to the next speaker, we would like to share your short biography that early sorry. Please, the admin to share the biography of Dr. Muhammad Fazrul Hizam, because you didn't give us chance to share before your presentation. Sorry. Yeah. Dr. Muhammad Fazrul got the bachelor from University Malaysia Terengganu and obtained master in University Teknologi Malaysia and doctor in the Prince of Songkla University. And Dr. Muhammad Fazrul Hizam expertise is fishing gears and fish ecology. And he also has many networking and research collaboration with Prince of Songkla University, Chulalongkorn University, KZ SART University of Hokkaido, Japan. And his research focus on impact of selective fishing gear to fisheries, feeding ecology of marine communities, fish behavior toward intrinsic and extrinsic factors, and management of fisheries resources. That is all for Dr. Muhammad Fazrul short biography and or profile. And before we go to the next speaker, I would like to correct the misspelling of Dr. Yan Evi Mabel. Yeah, because I misspell. It's very difficult to say your name. Sorry. Thank you very much, Dr. Muhammad Fazrul Hizam. So I would like to invite the third speaker, Mr. Johnny DeAng. He's a co-founder, Chief Innovation Officer, Cogner AI, Artificial Intelligence and Data Science, Melbourne, Australia. And he will present with the theme State of the Art machine learning and data sciences for marine and fisheries sustainability. We welcome Bapak Johnny Diong. You have 20 minutes, please.
Yeah, thank you, Dr. Ashmi. Let me just organize this. All right, first of all, good morning to Your Excellency, Dean of Faculty Marine Science and Fisheries of UNHAS and Director of UMT, and to honorable committee members, honorable speakers, distinguished guests, ladies and gentlemen. So my name is Johnny Deong. I'm a co-founder of Cogna. It is a data science and artificial intelligence company. We're based in Melbourne, Australia. When people in my network found out that I was going to speak on this esteemed marine and fisheries forum, their immediate reaction was asking me what do I know about marine and fisheries. They, of course, are right. Marine and fisheries are not my domain. However, from working in a project that stemmed out of a challenge presented by Australian Institute of Marine Science last year, which I will speak about in a minute, I voluntarily went through a piles of marine and fisheries research papers to augment my machine learning, especially computer vision knowledge. Now that apparently just barely enough credits, and hopefully I can contribute some points of view in the discussion today. With the evolution of AI and data science, we will see more of these collaborations between people like us in AI and data science field with the domain expert like you, teaming up to create a breakthrough AI solution for various domains, including marine and fisheries. In fact, in the last two previous sessions, I have already seen, Professor Bell talked about sponges and the coral reef reconstruction. That's obviously one of the area that's in the trend of machine learning and data science for us to solve, as well as the previous one by Professor Dr. Hisham. That's the bycatch. I will try to touch that quickly. I have some few points on that one. The bycatch reduction is also a problem that we try to solve in the machine learning in the marine and fishery. Now, let me quickly go through the main topic of I'm presenting this morning. Let me share my screen. I assume we can see that?
Yeah, it's great.
All right, thank you.
But before that,
All right.
you started, would you like to us to share your biography, please?
Sorry?
We would like to share your profile and okay.
I don't have it here. Okay, let me. Okay, so what my whole life has been in the IT world, right? I graduated in Monash University in 1998. Since then, I've been working for mainly IT services organization, Anderson Consulting, which is now Accenture, and IBM. Before I set up my first company, Epistemic, which is doing a lot of IT architecture, solution delivery, and specializing in the cloud migration. And before I continue on setting up this Cogna, which is we are specializing in AI and data science. So all the around 28 years I've been in the IT services across many different IT technology generation. So that's probably just a quick brief on my careers. All right.
Thank you.
Yep, hopefully that's enough. So, okay, this what I'm presenting today is set of this is really aligned with the theme that we are having in this symposium, which is the sustainable development. And I will quickly just go through. I only have 20 minutes, so this is just a standard AI data science machine learning blurb. I will skip this. I'll just go to the main topic. So as I mentioned, we at Cogna has developed a machine learning solution, which stemmed from the Australian Business Research and Innovation Challenge, which is presented by the Australian Institute of Marine Science, AIMS. So what the problem, the challenge that has been presented to us is they have an existing system, which is based on the BRAF system. BRAFs, I think, I'm very sure you guys are very familiar with this. This is the baited remote underwater video stations. Just a brief of this is quickly, there's a two pole, they put a bait there to invite the fish come over. At the other end of the pole, they put a video camera to capture all the video. So this is the method that they use today to do the fish survey, and this is done very manually. All the recorded video will require a marine scientist to sit on and identify the fish, count the number of fish, measure the biomass of the fish, counting the abundance. So that type of work is done manually. So the challenge to us is how we can automate this. So we put our solution to AIMS, to BRI, and then since there on, we have been further develop our own solution independently. We have branched out from there, which is what I'm presenting today. So the main functions of the solution that we put forward, which is really initially the one that you have there in the center of the screen, we are really just delivering a capability to do the fish survey. So there are 4 points that we need to capture. The appearance of the fish, which is what it means is we have to work with the 1 hour length of video. As you can appreciate, there will not be fishes in all the frames of the video. So the first task is to identify where the fish appear in one video session. Once we identify that the fish in the video frames, then we have to identify the species. Then we calculate the abundance as well as measuring the biomass. So those are the main objective of the solution that we delivered. This is all. We try to move all this manual work into the automated workflow. That is the challenge that we had. Now, as I mentioned, we make that all autonomous. So what we are proposing here was a machine learning solution. We have a continuous learning algorithms which I will walk through quickly in a minute. And then we do the auto labeling to identify the fish, which we populate into our main database, or our production database. The design that we have is we induce a lot of collaboration in the solution that we put together. We are developing the marketplace add-ons. We include the content adapters because initially we are only taking the BRAFs video, but then we have further on extending the multimedia, the input source that we can work with. We have a collaboration. We use the public domain sources like a Fish for Knowledge. If you are familiar with, there are a lot of like sort of encyclopedia of the species that's done by one of the marine research project before called Fish for Knowledge. We work together with one of our partner to have access to the data sources. We also we also built an API gateway to provide access to external parties to our information, as well as a human user portal, and we are planning to do our mobile devices as well. The user groups that we cover, in this solution is the marine fishery scientists, to the people like you. We also cater for the traditional owners. That's what we call them here in Australia. As well as the citizen scientists. So this is just anyone that is interested in the fisheries. They can contribute information as well as consuming information from our data source. The public users, this include education, student, teachers, as well as the machine-to-machine user group. So this is basically automated integration with other systems. Within the data science world, the data management obviously is one of what we are focusing on. We collaborate with a lot of public collection, as I mentioned, the Fish for Knowledge, for instance. We also do a lot of curation, collecting a lot of different information and curate the information. This is the one that we are going to share with the external group. Because a lot of the information that we have, a lot of metadata, that probably doesn't make sense if we just expose it that way. So there is a level of curation that we need to do before we can share the information to external people, to the public. We make our referential database for building our knowledge graph. Then the way to distribution through the web, email, and a lot of different distribution mechanism. Then we do enrichment of the data and the analytics of the data. That's all the functions that we have in our solution. The next part is the enterprise services. This is probably the boring part, but it's important because to build our solution that have enterprise grade, we build a strong security, logging and auditing, monitoring of the system, capability to integrate with a lot of different systems, the reporting, and then the notification system. All this solution is built using a user-centric with a straight-through processing with the intention of to make this as autonomous as possible. So we minimize a lot of manual steps to make it user-friendly. We built a very responsive user interface so that when human interacting with the system, they know what's happened, they understand intuitively how to use the system. And of course, every action there, critical action, is auditable. So that's on the high level, the function of the system. I need to move faster, I think. This is the solution architecture that we have. I will not go through in detail but I will touch quickly what it is. The bottom part here, this is the autonomous learning that I was talking about. So we built the system that's continue on learning based on the new information that we fed to them. So the data that coming through this learning system, it can comes from our initial production data, the clean data. That's the main source, but it also comes from the internal marine and fishery scientists that provide the information, do the data augmentation. They all fed into the learning system. Right, and the second part on the middle here, this is what the inference services. This is the key components that are really building, really doing all the function that we mentioned about detection, measuring, and measuring the biomass, calculating the abundance. That's all happen in this middle section, the autonomous inference services. So that's all happen automatically. Just a quick workflow. So we get the data from the graphs, then the information, the video is adapted into the common model that our system can read, and it's run as an inference golden model. So this is the model, the algorithm that built that identifying the fish, calculating the fish, and doing capturing all the metadata. This the model here is actually improved over time. As we said previously, there's a learning system down there that we built. Every time the system go through successfully the cycles, the capability to inference the fish, the organism, improve. We promote that into the models, which then can, based on the certain automation rules that we built here, it can become the golden model, the next golden model. So basically, the system is improving learning with the time, with the information that we fed them. Then that is the key components of our solution. As I mentioned, there's a production data that we are going to expose as a public content services. This is the curated data source, because there are a lot of information that we have in our production data, but not all we are going to expose in here. And then this one, the information will be exposed through external gateway for other consumer to use, human and machine. Externally, we're also going to build the marketplace with all the different type of media, different type of data set, different type of algorithm, different set of automation. So this all making the full set end-to-end enterprise system that we built for the fish survey system. Just quickly, on the deep learning algorithm itself that we are building. So there are two major components here. I start with the first one on the top. So there are two major components in our inference system. The first one on the top is, as you can see, we basically do the input is actually the image of the raw video. Sorry, it's not the image, the video of the graphs that we receive. We run this through an open source object detection system, which called YOLO version 3. YOLO is you only look once. SSD is the object detection algorithm that's available as an open source. So the intention here, we use an open source. That is the known and the latest state-of-the-art algorithm to identify the fish species. That is just one part. The second part is what we really built. So we use this two paths to identify, to inference the fish, basically to increase our confidence of how our machine learning can identify the fish. So the one that we are building, the input is the same. We take the image frame. We use a Gaussian mixture model. Gaussian mixture model is what in the machine learning world called unsupervised learning. So it's basically grouping the pixel, the image in the frame or in a video to the main intention there. For instance, we are going through differentiating the background to the front, to the foreground. The foreground is obviously the object of what we are interested in, and then combine that with optical flow to identify the movement of the fish. Then we know that's actually the fish that we want to do an inference on. It comes through this is the deep learning algorithm that we use for each of the layers that's identified in the Gaussian model. We do the classification, and the output is the same, the bounding box of the fish species, and with all the metadata. And those are the output that we do the comparison. The reason is we want to increase, or we want to detect what is the confidence level of the machine learning prediction that we have. Depend on the similarity or dissimilarity of the two output, then that's basically defining the confidence. Then there's a biomass calculation. This is just basically mathematical calculation. the rules. What interesting here is we also introduced a knowledge graph. This is the main value adding of our solution. This knowledge graph, as I mentioned, is basically take all the information from many different source system. For example, when we do the inference in the let's say the coast of Victoria or South Australia, we identify a fish, and then we know from the knowledge graph that that specific fish probably live in the tropical ocean and marine system. That's probably reducing our confidence level. So this knowledge graph is basically a collection, a network of the information that bring up the statistic of the profile of a specific species. Based on that, then the output, if the output is the data, the information about the fishes that we do the inference on. This is just a quick screenshot of what it is from the video stream. We identify the fish, as I mentioned. Then, based on that specific bounding box, we identify the species of that fish. This one here, this is an example of the Gaussian model that we use to identify a specific area of the fish. This is how the computer vision identify objects in the Gaussian model. There are a lot of challenges here. Sorry, how much time do I have, Dr. Ashmi?
Yeah, 5 minutes left.
5 minutes, all right, okay. So challenges, I mean, obviously there are a lot of challenges in developing our solution, and we're still working through a lot of this. The technical challenges, there's a complexity and variance underwater video data. As you can appreciate, the underwater video, the lighting is not perfect, not as, it's very challenging to sometimes to capture the fish, to detect the fish movement, orientation of the fish. It's always challenging because of the lighting of the video that we have underwater. We have a challenge with the low resolution video and the long distance field of view, especially in the BRAF system. Sometimes we know there's actually fish, but it's the distance is very far from the video camera. It's very hard to identify or infer the information that we need to capture from that specific frame or species of the fish. Now, the third one is the limited access to labeled data. This I think is a generic, a common problem within the machine learning and data science field. It's not only specific for this fish survey. We always have a challenge in identifying, getting a good data that has been labeled, because I mean, in order for machine to learn, we need to basically educate the machine, right? So the way we educate it, we provide them with the information, the ground truth. That's what the terminology we usually use in the machine learning. So the ground truth is usually very challenging to find, not only in this fish solution, but across the machine learning. This is the common problem. There are a lot of information, a lot of data on the internet, but the reliability of that data, is always the challenge. There's a functional challenge, a different type of fish species, like there's a benthic species living in the seafloor. That is very similar to the rare species. We probably don't have a good ground truth of that, so it's very hard to identify that type of fishes. There's a sedentary species living in the cavities of coral. That's sometimes we are overestimating or underestimating. It's probably more overestimating, because the same fish usually hide inside the corals and coming out swimming again. That's basically double counting. So it depends. There are different type of species and different challenges in that. The species swimming behaviors, right? So the fish move, it's very different to human. I mean, if we compare this to the solution in the machine learning, like a self-driving car, in the self-driving car, the autonomous driving car, we usually need to identify human that crossing the street, right? Now, human movement is very predictable into the common direction. It's different from fish. Fish can swim across 360 degrees direction. We need to identify those, and especially challenging in counting the abundance of it for that, and identifying the species. The natural phenomena, I talk about the lighting, the sunlight, it also making the video become very hard to infer. There's other administrative challenges. I will not go through that. I just want to quickly mention, this is what I'm saying. So machine learning in the marine and fishery, they are very relevant. I mean, we should be working together. I mean, as I mentioned, I've been listening to Professor Bell earlier. I mean, he talked about the sponges, but the destruction of the coral, for instance. So with the similar technology here, we have, there are some work around identifying, start to recording the benthic substrate with the intention to reconstruct the damaged coral reefs. Based on the computer vision, we can identify the species that required for the specific area. The previous speakers also talk about the bycatch, the problem with the bycatch. There's a solution in the machine learning. Again, the first one is in the computer vision to identify the bycatch that we should not be capturing. So there is the computer vision, and there is the echo sounder solution, the echo sounder version of solution to that. We based on the echo sounder data, we identify what type of a fish in the specific area where we should be directing the fishermen to do the catching. That's the implementation to that in the tuna percent fisheries. And the last one here, it's a very similar to our solution, but this is the implementation into the aquaculture or aqua farming with the machine learning solution. With the computer vision underwater, we can do the biomass measurement, disease identification in fish compound, measuring the optimal feeding amount and schedule. So the point is, this is the machine learning data science is very closely capable of resolving a lot of problem in the marine and fisheries, especially with our intention to move into more sustainable marine management and then fisheries procurement. That's probably from me, Dr. Ashmi. Back to you.
Thank you very much, Mr. Johnny Diong. We believe that your background with the machine learning and data science can encourage us to have a very good collaboration between a researcher and your company. And ladies and gentlemen, we just to inform you that we UNHAS and Dr. Johnny Diong, the Cogner AI, will have MOU, Memorandum of Understanding, to how use the data machine learning and AI to implement in the research on the related to marine science and fisheries. And yeah, we can see the short background, biography from Johnny Diong and his work experience and work background will, I think this is very good experience and skillful to support us to use artificial intelligence and machine learning in the research. Before
Looking
we
forward to that.
Yeah, thank you. Thank you very much, Pa Jonidi. So this is, we move to the last speakers. For this first sessions, we will have for the invited speakers, and the second session also we will have the another four invited speakers. And ladies and gentlemen, distinguished guests, we also welcome you to give some questions directly to the chat room, so the speakers can directly answer your questions. And then after we finish the four invited speaker, we will have panel discussions with the speakers. So we Now we move to the next speakers. Associate Professor Dr. Satoshi Asaoka from Graduate School of Integrated Science for Life, Hiroshima University, Japan. And let me read his profile or background. He's a professional environmental scientist interested in the water environment with a broad perspective: spring water, river water, seawater, wastewater treatment, restoration of coastal sediment, etc. And he got his Master of Engineering on Water Quality Control from Graduate School of Engineering, Toyohashi University of Technology, Toyohashi, Japan, and a Doctor of Philosophy in Grade School of Biosphere Science, Hiroshima University, Higashi, Hiroshima, Japan. And he has many working experience. He's from 2009 to 2012. He got a postdoctoral fellow in Hiroshima University, Japan, on geochemistry, green chemistry, and 2012 to 2020 as assistant professor in the Research Center for Inland Seas, Kobe University, Japan, on the environmental analytical chemistry, environmental remediation. And 2022 present, he's an associate professor in Graduate School of Integrated Sciences for Life, Hiroshima University. And he also got has award on 2018. He has been awarded the Commendation for Science and Technology by the Minister of Education, Culture, Sport, Science and Technology, Japan, Development Category, Ministry of Education, Culture, Sport, Science and Technology, Japan. Please welcome Professor Satoshi Asaoka.
Thank you, kind introduction. I'll share my slide. Okay. Is it okay? Can you hear? Can you hear and can you see my slide?
Oh, please, your sound is still slow.
So,
So,
yeah, thank you very much. And thank you for kind introduction, Asumi. And first, we thank Dr. Asher Harm and Dr. Najmuddin and Dr. Jamal, organized committee, for inviting me to prestigious symposium. Today's my presentation is entitled Remediation of Coastal Marine Sediments using Granulate Coal Ash. Japan has 88 enclosed water bodies. In this presentation, I'll introduce our environment remediation project conducting Set Inland Sea, Japan. The Set Inland Sea, which is the largest enclosed seas in Japan. The sea is located in western part of Japan. You can see a lot of beautiful sceneries and a lot of islands. The protected nature of the Set Inland Sea is ideal for aquaculture activities. This figure shows horizontal distribution of hydrogen sulfide concentration in sediments. This bar indicates sulfide concentration in sediments. The unit is milligram per gram. The hydrogen sulfide exert negative impact on aquaculture organisms, even at low concentrations. Therefore, in Japan, the concentration of sulfide, including AVS, is regulated at less than 0.2 milligram per gram under the aquaculture criterion to maintain a healthy aquaculture activities. However, as you can see, about 1/3 of the set industry area do not meet the sulfide concentration for aquaculture. Here, this slide shows the current strategy of environmental remediation for eutrophic enclosed seas. First, reduction of nutrients dissolution from sediments to suppress primary production due to internal cycles. Second, the reduction of sediment oxygen demand to prevent formation of poor oxygen water mass. And third, the remove toxic hydrogen sulfide in sediments to maintain a healthy aquatic environment. So this slide shows the approach of remediation for sediments. First, it is necessary to characterize the environmental remediation agents in laboratory experiments to clarify the chemical composition and adsorption performances and both environmental remediation performances and risks. And you can get useful parameters to simulate mass flow or propose optimizing strategies. And second, semi-field experiments. Semi-field experiments conducted monitor to environmental remediation processes using a water tank prior to field experiments, because semi-field experiments tell us both advantages and disadvantages of testing agents to remediate aquatic environments under the controlled environmental conditions compared to natural environment. And to design the numerical simulation model is a powerful tool to calculate mass flow and able to estimating the removal effect, remediation effects, and to propose optimum remediation strategies. And the final stage gate is field experiments. In the field experiment, we should cross-check the field data and estimated the obtained from the numerical simulation model. So in this presentation, we will introduce the environmental remediation experiment using a granular coal ash. By the way, what is the granular coal ash ZCA? This slide shows illustration of a coal thermal power plant. The fly ash is categorized into two fractions, bottom ash and fly ash. The bottom ash is collected under the boiler, and the fly ash is collected from waste gas treatment processes, such as denitrification, electric dust collector, and the desulfurization processes. About 85 to 95% of coal ash is fly ash. So this is why we use the fly ash. The GCA is produced by the granulation processes of fly ash by adding cement, accounting to 50% of the final products. The photo shows a scanning electron microscope image of a GCA. When the coal ash mixed together with cement, the silicon oxide and aluminum oxide in the coal fly ash react with calcium hydroxide and forms a new crystal phase, such as calcium silicate and calcium aluminate. This particle image is fly ash particles, and the new crystal phase is filled in this gap, like this. This table shows the chemical composition of GCA. GCA is mainly composed of inorganic elements such as silicon oxide, calcium carbonate, and aluminum oxide and calcium oxide. And the organic carbon is less than 3%. And heavy metals is zero compared to the coastal sediment in Japan. Let's go to the results. This is one of example of a laboratory experiment for removing hydrogen sulfide solutions. X-axis contact time and the y-axis hydrogen sulfide concentrations. The initial concentration is 8 and 80, which represents the hydrogen sulfide concentration in a segment in enclosed water bodies in Japan. And testing condition is described here. And the blue circle is controlled without GCA applications. And red square is with a GCA application. As you can see, the hydrogen sulfide significantly decreased within 2 or black value indicates control container without GC applications, and the blue flow is a GC application containers. Unit is milligram sulfur kilogram wet sediment per days. As you can see, the 0.65 of hydrogen sulfide is generated by sulfate reducing per day. And the control container, most of hydrogen sulfide oxidized to elemental sulfur with consuming water, dissolved oxygen in water columns. In contrast, GCA application container, the hydrogen sulfide generation rate is the same. However, most of hydrogen sulfide is diffused to the GCA on the sulfurs and adsorb and oxidation. Consequently, the hydrogen sulfide oxidation is reduced 0.044 milligram S per kilogram weight per day. So applying GCA, HH oxidation decreased 90.7% from the controls. And finally, field experiments carried out. This slide shows a field trial for removing hydrogen sulfide in sediments for water using GCA at Black Lake in Japan. Here is experimental condition described here. The GCA is scattered over the organically enriched sediment in Shimane Prefecture, Japan. Water depth is 3 to 3.5 meters. Area is 110 meters and 140 square meters. Bed thickness is 20 centimeters. Particle size of GCA is 3 to 4 millimeters. This is the results. This is a time course of the hydrogen sulfide concentration in sediments for waters. Two control site is established. Control one is a natural environment without any treatments. And control two is the sand capping. Sand is scattered with the, sand is covered with the organically enriched sediments. This is in Japan is a conventional environmental remediation method to suppress a dissolution of nutrients from the sediment to water column and suppress hydrogen sulfide formations. The blue diamond is a GCA application site. So as you can see, the hydrogen sulfide in sediment of water decreased significantly compared to controls. Conclusions. One is the GCA removed the hydrogen sulfide effectively through oxidation. And the second, GCA from coal electric power plant is a promising sustainable materials to remediate eutrophic enclosed water bodies. And third, the proposed simple remediation method can also promote recycling consciousness within society. Acknowledgement. We thank again Dr. Ashraf Farm and Dr. Najamuddin and Dr. Jaham Organic Committee for inviting me to prestigious symposium. Finally, I'd like to extend my heartfelt thank you for all our colleagues and thank you for your attentions. Thank you very much.
Thank you very much, Associated from Dr. Satoshi Asaoka from Graduate School of Integrated Science for Life, Hiroshima University, Japan. Distinguished guests, ladies and gentlemen, now we are having this panel discussion for invited speakers. Please raise your hand if you want to have questions to all speakers or to the give to the chat room, and the invited speaker can directly answer. Any questions? Yeah. This is please raise hand. Yeah, I think in the chat room all the questions have answered from Professor James Bell and
Hello, good morning.
yeah. Good morning. Yes.
I'm Umbu from East Nusa Tenggara, Artawacana Christian University.
Dari mana?
I want to ask my question for Mr. Satoshi Asoka. The question for Mr. James already be answered. The last question for Mr. Asoka. Can I do it, please?
Yes, Pak Umbu Dewa.
Okay, my question is, you are talking about the remediation. I want to ask about what combination that you use or respective techniques can produce maximum benefits and increase remediation efficiency to deduct the sediment. What combination that you use for deduct the sediment in your research? And did you use some bacterial for deduct the remediation, something like that?
Okay, your question is from James Bell, right? Sorry, for Associate Prof. Satoshi. Yeah.
Question for Mr. Asoka.
Okay.
The last speaker.
Okay.
The question for Mr. James already be answered.
Prof. Satoshi Asoka, could you please reply?
Thank you for questions. Combination of bacteria, your questions. GCA is, I share my slide. So.
My question is already that I already put in the chat room. Maybe you can read that.
Chat room, yeah.
Yes.
GCA is calcium concentration is high. The sediment pH is increased. Maybe sulfate reducing bacteria activity is suppressed. The sediment pH over the pH 9. So this is why the application of GCA both chemically and biologically reduce hydrogen sulfide. Is it okay?
Pak Umbu Dewa?
The combination for the CAO, something like that. And then also I want to ask that, did you use some bacteria?
Yeah, he used that. He asked about the bacteria you used.
Yeah. Your suggestions?
Yeah. Because I'm talking about the remediation. I know that there are some bacteria that we use for the remediation, something like that, sir.
Yeah, thank you.
Okay, if you did not use, thank you for that.
Thank you very much. Thank you for good suggestions.
Okay.
So, it's okay, Pak Umbu?
Yes, welcome.
Any questions?
Hello, Assalamualaikum.
Waalaikumsalam. Please mention your name. Okay.
Yeah, my name is Umi.
Yes,
Zakia
Umi Zakia.
from Brawijaya University. I would like to address my question to Dr. Fazrul. Is that possible? Hello,
Yes.
Dr. Fazrul.
Dr. Fazrul, are you there? I think maybe the the he's
it's not available, Dr.
yeah,
Fazrul?
he's not available, I think. Let's the other.
Oh, okay. Then probably I would like to have my questions to the second question to Dr. Asoka. Dr. Asoka, usually in our country, the problem of sediment, sediment pollutions occurred in estuary. Is that possible within, I don't know, maybe large scale to treat the river water? You know, in some part of Jakarta, I saw they put a sort of a filter, yeah, probably first mechanically, physically, and then the second should be like chemically. Now, is it possible to put this, what you call GCA, after the physical filter in the mouth of the river, so that the eutrophications in the coastal area can be reduced? Do you think how long it takes for this GCA to stand over there? I mean, when we put it into the second or the chemical filter in this treatment. Thank you.
Thank you, Dr. Umi.
Thank you very much. Good questions. Oh, I share my slide and so here, okay. Shoot. Maybe we use the here. This is our the GCA at the sorption site is regenerated under the oxygen conditions. Maybe theoretically permanent, effective permanent is continued. The filter media is also I check the removal performances, brackish and wastewater. It's how can I say? It's effective to reduce concentration of sulfide. Already checked. Maybe filter media is good, and effect is maybe oxy condition is maybe lifetime is permanent. Do you understand?
Just a little bit. Yeah. Now, probably I didn't get it clearly. Okay. But
yeah.
Okay. I was thinking like probably we can put this chemical treatment.
Yes. Okay.
Into a filter that
filter media.
For river remediations.
Yeah, Prof. Asoka, Dr. Umi, Ibu Umi Saki asked about the possibility to use your technology to treat the river water, especially in Jakarta, right, Bu?
Yeah.
Using remediation.
It works. Yeah, it's effective.
So is it possible? Do you think how long is this should be replaced in every period of time or no?
Maybe permanent because theoretically permanent very long time, because oxygen condition is this adsorption site is regenerated.
Yeah. Okay, so it means that it should be like replaced every period of time. To make it possible. Thank you, Dr. Asuka.
Thank you very much.
Okay. So, Dr. Umi, Muhammad Yeah, Dr. Fasrul Hisam
I'm here.
now is in.
I'm sorry because
Yeah, yeah.
my internet connection is not so stable, so I'm sorry. Okay.
It's okay. It's okay. Thank you for your coming. Um, I sort of have experience, I guess, when I was in New Zealand while I was
Yeah.
there, James.
Yeah.
Um, I met yeah, company over there. They used to reduce the over there in New Zealand. Some fisheries, some fishermen used sort of modification in the what you call it, the trawl or the seine or whatever, by adding some windows so that the unwanted catch or the bycatch can sort of free from that windows.
Yeah. Yeah.
Yeah. Um, yeah, of course, it's probably a little bit different with what you uh, present just this morning. However, do you think that by using this kind of modification in the um, in the catch, what is it, in the net, is it still efficient?
Okay.
And
Okay.
my
Okay.
second question is, it seems like the what is it, the machine that you propose is quite expensive for the fishermen to have in their boat. Do you think that it can be simplified, or what is the efficiency of these facilities so that every fisherman can have this kind of machine in their boat? Thank you.
Okay. Welcome. Thank you, Dr. Umi. Okay. For the first questions, is it possible that we would like to deploy some excluded device at the fishing gear? For the excluded device, we can just use at the what we call passive fishing gears, such as porcine or tronet. But for the artisanal fishing gear, we cannot use that. So we have to modify the fishing gears by combining mesh size or change the material. For example, for the ghost fishing, if we lost our fishing gears, the fishing gear is still active to catch the fish. So right now I'm designing one material that they will what we call they will destroy by itself if they lost in 4 or 7 days inside the water. So in seven days, the material decompose itself compared to our previous one. That's one, and the other one I also have some collaboration with other university in Thailand and in Japan to design some frequency. We put it the device in the net, so certain frequency we reduce some kind of bycatch, such as sharks. Right now, we focus on sharks because we catch a lot of sharks and also and rays. Certain rays is underprotected in Malaysia, but they have some kind of value, so the fishermen still keep selling it. Okay, for the second questions, actually, for the echo sounder or sonar, they have two types of fish detection. We call one commercial echo sounder or sonar, and the other one is scientific echo sounder. But for the scientific purpose that I show it to you before, is scientific echo sounder that we want to collect the target strength. But based on my finding during my research, we use the acoustic imaging to tell the fishermen, especially for the deep sea, to go to catch the fish under 50 to 30 meters only, because the matched fish only habit in there, and the fish behavior is very unique. So we need scientific echo sounder to understand their movements, because they are distributed based on the habitat, seasons, and the food, of course, and the mating seasons. They are very, for example, race in Malaysia, the peak season is around May to June, but in other region it's not. They have different kind of distributions. So for example, the prawn in Malaysia, they peak around March to May right now. Okay, March to June, sorry. So we have to identify which kind of season they have, but Maybe in Europe they have 4 to 5 seasons. In Malaysia we have 3 seasons only. We have rainy, moderate, and dry seasons. We have three seasons based on the rainy data. So we just can separate our season based on 3 seasons. It's kind of separate the ecological part playing an important role for the fish behavior. Okay.
Thank
That's
you.
all from me, Dr. Min. Thank you. Thank you.
Thank you,
Can I add to that, Dr. Umi? So
Yeah.
yeah, Dr. Hisam. So with the bycatch trolling, as I mentioned in the last slide that I show, uh, it is actually there is some project happening at the moment. This is a combination between machine learning and IoT. So they are developing, well, the core technology of the identifying the bycatch is very similar to what we had. So within the troll, they basically get the video camera to identify any, of course, the video camera connected to the server on the boat that has been trained for species of interest, right? So for those species of interest, when the camera on the troll identify the bycatch that we want to avoid, then they deploy the door, the one-way door to release the bycatch. So there is that project happening at the moment. Of course, I don't think it is commercialized yet, but that is happening. Similarly, as I said, the image computer vision detection, as I mentioned, or as well as the using echo sound.
Thank you, Mr. Ong, for the
Thank you.
explanations.
Thank you,
Thank you,
Doctor.
Prof. James, as well. Thank you, Dr. Aska. Thank you so very much.
Okay, very good discussions. So there is another questions to Dr. Satoshi from Widya Stuti from UNHAS. Is the GCA composition has side effect specifically for benthic sessile organism? Dr. Satoshi, are you there?
Yes, thank you for questions. It depends on the application dose. We optimize the application dose by the simulation models to prevent a rapid pH change. The high pH is, as you can see, a negative impact to the benthic organisms. But a small application dosage is no effect to pH change. And some elements is, wait a moment, please. And I also share the slides. This is a dissolution test dissolved from the GCA. This is environmental regulated elements regulated under Ministry of Environment in Japan. And this is results, and this is a maximum limit of the criteria. If the concentration less obvious zero, the organism is no effect of these substances. This is why the organic environmental regulated element is obvious zero, standard maximum for environment. Is it okay?
Yes, thank you for the answer. So I think Ibu Widya Stuti, okay, yeah.
Okay, ladies and gentlemen, we still have 10 minutes left for discussion. If you have any questions, please, you can ask by raising your hand or just ask, because I cannot see clearly from this side, from my side, who's raised his hand. Okay. Or maybe between the speakers we'll ask, like, Pak Johnny D, would you like to ask something to Professor James Bell, or, yeah, or Dr. Satoshi?
Yes, I probably have a question quickly, Professor Bell. I mean, sponges, I mean, this is the first time I heard uh, from your very interesting lecture. So how is this different to the coral? Actually, I've been looking at some of the solution, as I mentioned again in my last slide, some research on reconstruction of the coral reef. How is that different? I mean, obviously, well, I need to go through more material before probably can make the question meaningful. But if you can quickly just give an overview of it is. By the way, I have a lot of time. We have lockdown here down in Victoria. So
Yeah, we're lucky in New Zealand, I guess, to be one of the few places where we can probably still hold conferences with real people in close proximity. That's a good question. I guess a sponge reef differs substantially from a coral reef because the primary organisms change. And one of the, I guess, key things about sponges is, although I think I said some things eat them, a lot of stuff doesn't. So sponges are really well chemically defended. So there isn't as much food. They also, the other thing that I kind of touched on in my talk is that there's a real difference in the level of habitat complexity created by an environment that's dominated by sponges. Lots of corals, particularly some of the acroporids and branching corals, provide a lot more space for organisms to live and provide those kind of ecological niches which support higher biodiversity. And reefs that are dominated by sponges don't do that. I guess the other key difference is, although some sponges have these symbionts in their tissues and are photosynthesizing, they're primarily kind of heterotrophic animals. They're feeding on carbon that's in the water column, whereas corals to a greater extent are more dependent on their symbionts and the photosynthesis or photosynthetes that they give. So the overall reef productivity of a sponge reef is going to be much lower, I think, than something that's dominated by corals. And that just means less biodiversity, less fish, less food for people to eat and resources to take and have.
all right, thank you. So do you think that is actually among the answer to like carbon dioxide processing? I mean, even from the surface.
No, I don't think sponges are going to contribute too much to that, I guess. I mean, it kind of highlights it's not and obviously it's not a great scenario if all the corals decline and something else takes over. In fact, it's a disaster. But I think it's important for us to explore, the scenarios of what reefs might look like, and in case reefs are looking like,
Yeah.
try and figure out. We have a number of people working with us developing kind of ecosystem models to have a look at what the overall system might look like on the Great Barrier Reef if sponges become dominant, such that we can work through those scenarios and hopefully, convince people a bit more that they shouldn't be doing these things that causes the declines. The interesting thing about some of the reefs I've worked on is it's quite often it's the local scale factors that drive the change to a sponge dominated reef. So particularly things increases in sedimentation and nutrients, which are things that can be controlled at that local level. So environmental managers do have the ability to pull levers to control sediment. It's not like kind of global climate change where everybody in the world needs to work together,
Right.
which I'm not particularly optimistic about humans doing. We don't seem to do well when we've got big problems like pandemics. We don't do a particularly brilliant job of handling those things. So climate change is probably even bigger than that.
Yeah.
Just a slower burn for that. But
Yeah. Yeah. In some of my research, I came across this solution. Well, it's again, as I mentioned, it's focusing on reconstruction of the coral reef. That's the scientists today start to identifying. I mean, they help they get all the images from the divers that going down, and we start to collecting the common database to identify the coral model in a specific region, and that's obviously becoming an input to the intention is to be able to reconstruct some of them by understanding the benthic composition for each region. Then hopefully we can rebuild that. That's among the things that happening in this in the machine learning work.
The kind of use of your technology as well in that kind of monitoring changes without people actually needing to be there. You know,
that's right.
You know, underwater robots swimming around instead of divers much more efficiently. You know, with
yes.
software to understand the corals and the differences between the corals and sponges, and do that much more than humans do, and more consistently.
Thanks, Professor Bell.
Thank you. Enjoyed your talk too. It's good.
Thank you very much, Professor Bell and Mr. Johnny Diong. So now we end the first sessions. So from committee would like to thanks to all the speakers. Thank you very much for today, for very nice and very important information and new insight of related to the research and technology for marine and fisheries science. And for the next session, we would like to invite 4 invited speakers, and let me say hi to the Professor Nicholas Paul, because are you joining now, Prof. Nick?
Good morning.
Yeah, you are there already. And before we start the second sessions, again, we would like to say thank you very much to all 4 speakers, Professor James Bell, Dr. Muhammad Fazrul Hisam, Mr. Johnny Diong, Dr. Satoshi Asaoka, for very nice and very important and very insightful knowledge and experiences related to your research. And we are looking forward to the research collaboration between you and the researcher from Hasanuddin University, especially from Faculty of Marine Sciences and Fisheries. Thank you. And for the next sessions, the second sessions, the first speakers will be Professor Nicholas Paul. Now, Prof. Nick is already here. And let me share the biography, please. Yes. Yeah, Professor Nicholas Paul is a marine scientist at the University of the Sunshine Coast in Australia. He leads applied research in development on seaweed for new product development based upon a platform of sustainable production. And he's also a project leader on international research for development projects funded by the Australian Centre for International Agricultural Research, focused on domesticating new strains, creating new culture techniques, and developing byproducts for farmed seaweed, working with government partners across the Indo-Pacific. Professor Nicholas Paul interest in Australia revolve around high-value products, including functional food and nutraceutical for human health, as well as bioactive for fish and livestock. So we please welcome Prof. Nick. Your time is 20 minutes.
Good morning, everybody. Thank you for the opportunity to talk this morning. about seaweed, and particularly about innovations and how seaweed can contribute to sustainable development in both Indonesia and Australia. And so I'm coming from the Sunshine Coast in Queensland in Australia, and I'd like to thank the organising committee and as well as UNHAS, Faculty of Marine Science and Fisheries, for the opportunity today. So thank you, everyone. So one of the things that we're often framing around seaweed, particularly at the moment, is all of the economic, environmental and social impacts that it can have. And it's such a nice fit with the 17 Sustainable Development Goals that I'm sure that everyone is familiar with. Today, I wanted to talk about two aspects of the work that we've been doing, some Indonesian examples as well as Australian examples, and particularly for Indonesia, focusing in on the end of a large project that's just wrapped up earlier this year on seaweed production and processing, and where we were taking a whole value chain approach to looking at production constraints and developing diverse products as well as social impact. And then the second part of the talk will be really sort of focusing on quite a different setting in Australia, which is much more sort of industry high value production, new discoveries, industry partnerships, and looking at patent development, commercialization for the university. And I'll touch on those types of things. So the first part of today was really to focus in on what was a 5-year Australian Centre for International Agriculture Research funded project that just wrapped up in January, and we're still finalising the reports for that. The project aim was to step back and take a scientific basis to look to modernise the seaweed industry in Indonesia, but at the same time considering lots of environmental and particularly social impacts with the farming as well as the processing of the seaweed. We had lots of different partners in that project. For those that don't know, ACR projects are government to government ones, so we were working directly with our counterparts in Indonesian Ministry of Marine Affairs and Fisheries, particularly a couple of research centres, the Research and Development Centre for Marine and Fisheries Product Processing and Biotech in Jakarta, the Seaweed Centre for Research and Development from Gorontalo, Loka Rumput Laut. We worked with a couple of the brackish water aquaculture development centres in both Takala there in Sulawesi Selatan, as well as Jumbati, UNHAS, of course, and the College of Marine Technology there, STITEC in Makassar. So lots of different people, more than 30 core researchers across the whole project. And I'm only going to give you a snapshot of some of the key things that we've seen here. So when we're talking about seaweed in Indonesia, for the most part, it's Kappa-Ficus alvarezi, so also known as cotton eye. Here's some of the seaweed that's being harvested by a farmer in Bantang Regency there in Sulawesi Selatan. So looking across the entire supply chain of seaweed, there's a number of different steps. There's the production, the farming side, either in suspended culture or also off bottom style culture. That fresh seaweed that's harvested needs to be handled and then dried, ultimately shipped to a seaweed processing facility where, for Kappa-Ficus anyway, the primary goal is to make carrageenan gel, so the hydrocolloid, which is an existing product used in all different types of food and beverage applications across the world. So from the government of Indonesia perspective at the very beginning of the project and scoping work, it was very clear that the effort should now be on value adding for the existing sort of cultivation of the species of seaweed and even the selection of new species. That value adding and processing should be done as close to the production areas as possible, so as close to the farm production areas, and hopefully a range of new products can be developed to enable the significant growth that's already happened in Indonesia with seaweed to continue on for the next decade. But that leaves a number of question marks around, okay, if this is the existing supply chain, then what can we start to do to understand new opportunities? And luckily with that very diverse team that I mentioned, we tackled aspects of the entire value chain for this project. there are a couple of objectives. The first, the three particular objectives. The first was to look at the value chain, so understanding knowledge gaps or technology gaps in the supply chain, as well as socio-economic aspects. So there was a lot of effort done, particularly by our biotech team in Jakarta, on the value chain of the large seaweed being produced, the Kappa ficus and Eucuma, which are made for carrageenan. Lots of different insights from the farmers, remember smallholder farmers at community scale, through to the traders that are facilitating the movement of the dried product and processed product around Indonesia and exporting, as well as the processors. Lots of different insights there. So how is this changing the production over time, and basically where is it occurring? Because sometimes we don't understand that level of detail. And on top of that, looking at the quality of the seaweed that's being produced, because some of the reports were over time that the quality was declining and degrading. And so what are the constraints to maybe make more valuable products out of it? So a comparison here is some differences in the color of the dried seaweed. A number of publications that resulted from that, and I'll just touch on one of them here as an example, especially when it comes to sustainable development goals, is trying to improve the margins, so the actual revenue in the upstream, as far upstream as possible. So certainly from ACR's point of view in Australia, when they're funding research, they're hoping to have significant impacts on the farmer and the livelihoods of the families, especially if they're farming families. And so some of the technical parts that the biotech team identified with the farmers by walking the chain, so being out there, was that there's some value that can be had by managing moisture content more clearly, as well as trying to be more consistent in their culture productivity. So within the existing framework of what they're doing, trying to slightly change the process to get more value, which is an important thing for production and the like. And so there are publications around this, as well as other types of warehouse systems and receipt systems, to trying to industrialize as much as possible the process there. Some of the more exciting parts, and including collaborations here with staff at UNHAS, was looking at the socio-economic benefits of seaweed. So some of the reports, especially for women from different parts of the world, were that the benefits to them might not be clear. And in fact, there are a number of negatives. So we were curious as to how Indonesian women in particular, what insights they had around the seaweed farming and possible benefits for that. And so we did this with some new sort of approaches for collecting the data, so game-based scenario, providing lots of opportunity for the women to contribute their knowledge and insights into the system. A couple of publications have come from that. The first, again, it's a collaboration between Australian and Indonesian researchers, including some UNHAS staff, Dr. Mariana Fakri, as an example, was looking at women's well-being and the household benefits. So what has the community perceived as being important about seaweed, which has grown so much in Indonesia to date? So looking at particular positive economic and social impacts, and basically there were a number of well-being factors that were linked directly to seaweed farming, and that also enabled seaweed farming, which included the extra income that families were able to get, meant that they could have more transport, which means motorbikes, better housing, and other types of basic needs in education. And these types of loops were something that was really sort of interesting that we explored further. But primarily, there were no negative changes reported, were mostly increased in life satisfaction. And that was, interestingly, in these seaweed farming villages, by both the women who would directly, their families directly engaged in seaweed farming, as well as those that were not directly engaged. They were actually also benefiting from the seaweed farming. So a great sort of idea, and we unpacked that further. There's another publication that's come out, will be coming out in some press in the journal Ambio, which is really understanding these feedback links, because it's quite important to know that if you, especially if you're a government and you're changing some aspect of policy that might influence this system is that there might be unrecognized impacts. And so knowing whether or not there are feedback loops for particular items in a pretty complex value chain is important, and understanding all the capability. And so this has developed much more and gone beyond just both seaweed farming and seaweed processing, the women that were involved. And there's a whole lot of very interesting social networks and transport angles as well, which are reinforcing the sort of the benefits there. So these feedback loops are important for seaweed production. And if we're talking about industrializing, for example, seaweed farming, what's going to happen to these very clear community benefits? So that's something to bear in mind down the track. At the same time as doing a lot of the socio-economic work, we were doing more of the classic technical work on the seaweed farming. So like all forms of agriculture, seaweed has a number of important production constraints that need to be worked through. Because it's relatively young in terms of the industry, there's still, even in Indonesia, a bit of a need to identify exactly what the species are that are being cultured, and particularly the strains, and using molecular techniques as much as possible for that, so DNA barcoding. And so we need to identify and make sure that we have very clear taxonomy on species. And once you understand that genetic diversity, then that obviously means that more can be done from a selection point of view. So that type of work was always in partnership with particular our partners in Indonesia across the country, both university and government scientists conducting workshops, so that at the same time as doing the work, we're also building the technical capacity as much as possible. So my colleague, Associate Professor Joe Zuccarello from Victoria University Wellington here at a number of different projects. And the great thing is that there's the ability to work obviously on the Indonesian context and provide publications that are suited. So we know that in the tropics, that's where most new seaweed production has happened in the last few decades, and will more than likely be the primary focus for seaweeds in the future. So we have to do this molecular work, and not much has been done to date. So there's a lot of discovery that happens, but there are a lot of techniques and methods that need to be followed. So the government scientists that we're working with really sort of valued those inputs, and were able to then turn that around and put it into a publication as well. So there's that legacy there, which once we have the correct identification of the seaweed and the storage and processing, the teams can then start to truly compare growth and production between different environments as well, across different locations over time. So pretty important for that, and sort of quite technical work as well. So that's very valuable work, but it is quite technical, so it does need new skills amongst the teams, and everyone really embraced that really well. What that led to, of course, were then follow-up publications where we're able to use those techniques that the team had trained to start to unpack this common name use of a lot of different seaweeds. So the trade names can be quite different from the formal taxonomic names, and it is really important to understand what species are there. And so having that updated taxonomy has proven really useful. It's also identified in places that there's very little genetic variation in some of the marker genes. And so surprisingly, though, lots of phenotypic variation. So we have the genetic variation of this one particular marker, which is used for taxonomy, coming in as saying the exact same haplotype being produced of seaweed across all of Indonesia. However, we have still this very clear phenotypic variation in terms of the color of the strains being produced, the shape and morphology of them from different parts of the country, and having different types of production. And only one other species of Capophycus that we identified of the 15 key production sites was actually a different species. And so that was an important distinction as well. So these types of stock assessment is really important, especially when it's linked to growth and production. So lots of avenues for more publications there. Taking that approach on, Remember, Indonesia is a very large country, of course, lots of islands across the archipelago, with production that happens from the north to the south and a lot in the east of Indonesia, and still some in the western side. But a lot of the production and strains the individual strains haven't been coordinated in terms of the movement. And so rather than trying to find new diversity or the like, there's this concept, Rani Pongmasak from Loka Gorontalo, Rumpel out there, is out there and with the team looking at these new species, trying to rejuvenate them and make sure that they can grow faster and have greater gel strength in that time. And so this work is again being published, so some pretty sort of standard types of approaches for how do you compare these seaweeds. People haven't done these types of assessments before, so the team travelled all over Indonesia, conducted standardized growth experiments at the farm sites, and then also brought those seaweeds back to a common garden, the seaweed estate here as it is up at Pangkep, just north there of Makassar. And so basically when you do that, you can have some really good scientific outputs, and the publications are starting to come out there as well. But most importantly, finding these superior strains. And so those new strains, the farmers are able to grow in places and times of the year that they haven't been able to grow before, which means that they can look to become more of their time as being used for seaweed farming rather than just a portion of it. So some pretty exciting work there for the seaweed. Last objective here, just to highlight some of the really clear innovations that are happening. This is the Research and Development Centre for Biotechnology in Jakarta. So rather than just drying the seaweed and using it or shipping it for processing, there might be things that can be extracted from the seaweed prior to the shipment for the use for carrageenan. And so this is one new product which is called biosalt. So it's basically taking the pigment and the very high or healthy salts that seaweeds are able to collect from the ocean. And so very high in potassium, for example, much higher potassium in the seaweed than in the surrounding salt water. So these pre-processing steps are able to actually develop some new high-value products that the farmer is able to do. Likewise, right around
sorry, Prof. Nick, you still have 5 minutes left.
Thank you.
Sorry.
The back end of the supply chain, after the seaweed is being processed, there's lots of areas for the waste material to be used, again sort of closing the loop on the circularity there. And so using the waste stream from the seaweed in agriculture or making bricks, again the team from Jakarta are doing that. And of course, last but not least, some new functional products. So high nutrition and high value applications for food that could be processed even locally at a small type of scale. And so we're expecting to see sort of recipe book will be published soon, which is using all the women's groups' recipes and promoting their activities there. So watch this space, very exciting. So on the conclusion side for the Indonesia project, we have this supply chain, and now we have lots of opportunities for small business. We have new types of carrageenan that are being processed based on pre-processing. There's conversion of the waste to new products, again giving more value directly rather than using the dried seaweed just for processing. It can be immediately used for functional food, and likewise the pigments and the biosalts, and last but not least these rejuvenated cultures so that production can be more sustainable. So pretty exciting all around there. My pivot here was really just the last couple of minutes was to highlight some of the more high-tech work that we're doing in Australia for your knowledge. And a lot of that is because Australian setting is that seaweed is almost not produced at all commercially, very small amounts, but lots of opportunity. So we're right back at the beginning where we're trying to find new species for different types of applications, collecting them from the wild, domesticating them. We're growing them. I won't show the video here, but we have a large production system where we're able to scale up the seaweed to get to a scale that it can actually be tested and harvest these and do it in land-based types of systems for control. Some of the work that you might have heard of, which is from work that I've been involved in, is using this one type of red seaweed called Asparagopsis, where we add a very small amount and it completely knocks out methane production. So this is really important. Methane has a heating potential 28 times that of carbon dioxide, and more than 10% of global greenhouse gas emissions are from agriculture. So very significant work, but we need to actually start farming the seaweed because it's not being done yet. So we're looking at lots of different types of seaweeds, making sure that we can collect a whole variety for use in the Australian context, often using our Indonesian partners for inspiration. But we're trying to take a bit more of a sort of high-tech approach, I guess, in this biorefinery concept. So rather than just having one product from the seaweed, can we actually have multiple ones, and primarily high-value ones? So the example that I wanted to give here is, rather than just thinking about cows, fish and fish health is so important for aquaculture. Disease is a really big problem. We can target these niche markets looking at immunostimulants, so boosting the immune system and the productivity. With some pretty straightforward data, we've been able to find that one type of seaweed, the Asparagopsis, is the same one that is responsible for methane reduction, is also boosting the immune system. And now we have 3 patents involved to do that, and that's only going to get better and better. So my final slides were really that there's this now new focus where we can start to really improve the economic outputs from seaweed at the same time as environmental and social benefits. And that's really the key for new clean green products in Australia or in Indonesia. There's lots of aspects around high value products, functional feed and ingredients, and making sure that we've got right partnerships there for taking that research all the way out to market. And the partnership will certainly be the key. This is my last slide, but just to highlight some of the work that we've been doing with UNHAS for the last few years. I've been part of the Seaweed Centre of Excellence there with the World Class Professor Program. We have a Memorandum of Understanding from 2018, and now joint publications coming out and more to come with Ibu Chitra and the team as well on the immune side. So thank you very much.
Thank you very much, Professor Nick. It's very nice you put my photo in your slide, and yeah, we are hoping we have a publication collaboration together. Yes, and then we now move to the next speaker, because now it's time for the second speaker. We would like to invite Ibu Dr. Nurjana Nurdin, STMSE. Please, I would like to share her profile. Please show. Yeah. Nurjana Nurdin, Associate Professor. She is now head of Research and Development Center for Marine Coast and Small Islands, Hasanuddin University. And she's also a senior lecturer in Marine Science and Fisheries Faculty, Universitas Hasanuddin. She obtained her doctorate in the Bogor Agriculture University, Marine Technology, and a master in Marine Technology, Bogor Agriculture University. And she also took postdoctoral program, Atmosphere and Ocean Research in Atmosphere and Ocean Research Institute, University of Tokyo, Japan. Next, please. And a field of expertise of Ibu Dr. Nurjana is remote sensing and geographic information system. And she has a relevant some careers. She's from 2000 and present. She's now a director of Research and Development Center for Marine Coast and Small Islands, Universitas Hasanuddin. And Ibu Dr. Nurjana will present today with the title Spatial, Spectral, and Temporal of Image on Small Island Ecosystem Indonesia. Please welcome Ibu Dr. Nurjana.
Okay, thank you, Dr. Rasmi Chitra. Thank you for the organizer committee for inviting me. Assalamualaikum warahmatullahi wabarakatuh. Good day, everyone. I am very happy to share all of you about the remote sensing data and information on small island from the spatial, spectral, and temporal perspective. I will present some of our research about the spatial data and information. This is the collaboration of remote sensing laboratory with marine biology laboratory, ecology laboratory, and oceanography laboratory in our faculty, Marine Science and Fisheries Faculty, Hasanuddin University. There are some points would like to share. First, about the geospatial information of small island. How about the spectral library of substrate and benthic community on Spermende Island? And the information about long-term changes of coastal habitat on small island. And the next about the new technology and the new technology using unmanned aerial vehicle data in geodatabase system in a small island Spermode. This picture is describe what is the remote sensing. Remote and monitoring the physical characteristic of an area by measuring its reflected and measuring its emitted radiation at a distance, of course, typically from a satellite. Object, energy, water column, like this picture, atmosphere, sensor, and satellite are remote sensing subsystem. If you see this, the energy, electromagnetic wave, visible band are used for mapping habitat in coastal water under the sea. In the ocean, a blue band reaches the deepest, and the red band is attenuated in the shallowest depths among blue, red, and green band. We will not talk very specific about the wavelength. This slide, this is the map of Bermuda Archipelago since 2009 until now. We have carried out spatial analysis and field survey and store data for about 54 small islands in this area. We divided the small island into three parts according to their perpendicular distance from the mainland. The mainland is the orange color here, and we try to divide it based on the distance. The first part, small island located in this area, small island located in the less than 10 kilometer from the mainland. And then the second part, small island located at the distance of 10 to 20 kilometer from the mainland. And the last part is small island located about more than 20 kilometers for the mainland. We can share this table. This is a list of island names and the distance from the mainland. Most of these small islands are inhabited for about 40 islands, and only 14 islands are uninhabited. So it's important to identify the damages of coastal habitat ecosystem by human impact to mitigate the adverse effect and to realize a sustainable development SDGs of a small island. How about the data, ground truthing, and spatial analysis? We This is a list of satellite data commonly used for coastal habitat observation. Some are free access, like Sentinel, Landsat, And then some of them is commercial. The predates, quit bird spot, what view. It's very important, I think, to determine the geographic information, the position of ground routing site as precises of possible, because they are used for classification and evaluation of classification of pixel into bottom substrate type. We need an instrument for ground routing, the base instrument like a GPS, a logger data, and a specific GPS for high resolution. Ground routing was conducted to determine actual habitat distribution by taking picture with commonly with waterproof digital camera. Time synchronization of GPS was done, and the camera was automatically linked to location coordinate of a picture taken in situ. We have carried out several spatial analysis, namely supervised and unsupervised classification, OBIA, and then we use some algorithm, depth invariant index, for example, and then we try to link image and field survey data, and try to linking imagery with the anthropogenic data. We would like to show how we develop the spectral library. We have developed spectral library through their measurement of optical properties of substrate and benthic community using underwater spectroradiometer since 2009 until now. And we already have a spectral signature about 213 objects consisting of healthy corals, algae, macroalgae, and seagrass. This spectral library is a basic reference to recognizing and identifying a substrate and benthic community in coastal habitat. This is our reference to produce accurate map. It means knowledge on the spectral separability of benthic object is very useful for designing and optimizing for coastal habitat mapping. Some statistic analysis we use to found the best discriminate spectral between benthic community in shallow water. For example, if we saw this, we use a correlation analysis, cluster analysis, derivative analysis, and discriminant analysis. The results show that some of live coral have very high similarity with dead coral, with algae, such as Porites sp, as you can in this dendrogram in the red line. This means that we must be careful in mapping coral reef, because between live coral and dead coral covered with algae is difficult to distinguish, especially when using low spatial and spectral resolution. And then the result will overestimating or underestimating. This is some statistical analysis we're using second derivative spectra to discriminate between live coral and dead coral covered with algae. We would like to find the best spectral discriminate between this, and then the result, a beta discriminate, was found at 539, for example, nanometer for green live coral and deep covered with algae. It means if you would like to discriminate between green live coral and green deep coral covered with algae, it's good to use 536 nanometer. Next, we would like to, this is a It's the same analysis, statistic analysis using second derivative to result the best discrimination between live coral and dead coral covered with algae. The next slide, we would like to share about the information long-term change of coastal habitat on small island. This is one of the result of image analysis on inhabited island and uninhabited islands. This graph show the dynamic of an island, a populated island. Uh, we saw the change every 3 years from 1972 to 2016, some of 2019. We see this, the largest of dynamic change of bottom substrate is indeed coral category in period 1981 to 1990, and from 1996 to 1999, and from 2002 to 2005. Maybe we discuss this is the related, very related to El Nino and bleaching even, and relative stable in last three period 2008, 2011, and 2030. This is the largest seagrass area in the Spermonda archipelago in one of island. The name is Kondong Bali. The result indicated that decreases if in live coral area resulted in increased area of deep coral with algae and rubber during the 44-year period. A good phenomena because in this island, seagrass tends to expand every year. This is the simulation how the changes of habitat, coastal habitat. The next, this is the information about the uninhabited island. So the same dynamic change trend, the tendency decrease on island during 1972 until 2013, 2016, and 2009. This is the result. It's the same, I think. And then we already analysis the accuracy less than 80%, only 75%. And next we saw about in situ and remote sensing data we combine with post classification and geographic information system. Post classification results show the location area. If we see the red circle here, sorry. If we see this, the red circle, the greatest threat to coral reef on this island is damage caused by the activity of people who come from other island around that island.
Sorry, Bu Dr. Nurjana, we still have 5 minutes. 5 minutes left, please.
Experience changes from live coral to dead coral. If you would like to know where the place live
Sorry,
coral
Ibu.
become to rubber every 2 years.
5 minutes left, please.
Classification we can show as clearly. Thank you. This is the using high spatial resolution UAV on coastal habitat, small island. We already exposed almost more than 45 small island. This is very good, very nice imagery with spatial high resolution 4 centimeter, and then we can count how many houses in the small island. And then now we analyze coastal habitat using UAV imagery. And you can see this boot, very small boot. You can see this, the red color and another habitat. And then we analyze this too using OBIA method to mapping the micro atoll in the small island. And the last, we build the geodatabase system for spatial dynamic on coastal habitat. We have a big data for small island, which is updated continuously, and then we keep in this geodatabase system all data from field survey and spatial analysis are stored in this geodatabase system, and anytime, if needed, can be displayed and updated. And the last, this is the geoinformation small island spare model. This is approximate quantitative information about the small island of Spermonde. If you would like to know where is the largest land island and how wide is it, this is the information, more accurately. If you would like to know where the largest seagrass area in the Spermonde Island, where is the widest living coral, where is the least deep coral, and where is the less rubble from this geodatabase can give the information. The last conclusion, it's important to re-management of sustainable development SDGs during the pandemic and after the pandemic for coastal area. I mean, coastal area need micro data, not macro data. Accurate data must come from the lower level, from the research community, from the university in their respective region, and ideally, it should be integrated by the Ministry of Marine and Fisheries Indonesia. I think it's our data and information. Hopefully, it will be useful for researchers and the government. There are many things to do there, increasing the capacity of small island and coastal community. They with us. Thank you very much. My acknowledgement to the Ministry, Technology and Higher Education, Environmental System Research Institute Indonesia for supporting RGI software license and cloud capacity, the National Institute Aeronautic and Space 8, and thank you for all.
Thank you very much, Ibu Dr. Nurjana, for very nice presentations today. And we now move to the third speakers. The third speakers will be Dr. Judith Weiss. Dr. Judith Weiss from Rutgers University, Newark, New Jersey, USA. Are you still in the line, Judith?
I'm here, but there's a presentation.
Yeah, I know. Okay. Okay, just want to know that you are available now. And let me...
And I'll be taking questions, but the presentation is recorded.
Yeah. We will share later. Okay. Thank you. And before we start to watch the video presentation from Judith Weiss, please share the biography, short biography from Dr. Judith Weiss. Dr. Judith Weiss is a professor emerita of biological sciences at Rutgers University, Newark. She got bachelor degree from Cornell University and master and PhD from New York University. The research focuses on estuarine ecology and ecotoxicology, and she published over 250 reviewed scientific papers, a technical book on marine pollution, and several books for the general public, including one of marine pollution. And Professor Judith Weiss also interested in estuaries and salt marshes and their effects on organisms, populations, and communities. She also a fellow of the American Association for the Advancement of Science, and she was a congressional science fellow with the U.S. Senate and a Fulbright senior specialist in Indonesia. Yeah, Judith was been in Indonesia in UNHAS on 2006, I think. Yeah, Prof. Judith. Yeah, and Professor Judith Weiss also has many experiences, work experiences. She's an advisory committee for US EPA and the National Research Council, National Sea Grant Advisory Board, chairs the Science Advisory Board of the New Jersey Department of Environmental Protection, co-chairs the Science Technical Advisory Committee for the New York, New Jersey Harbor Estuary Program, United Nations Environment Program as a lead author of the World Ocean Assessment 1 and 2, author of the Intergovernmental Panel on Biodiversity and Ecosystem Services, chair of the Biology section of A3AS, board of the Society in Environmental Toxicology and Chemistry, the Association for Women in Science, and the American Institute of Biological Sciences, of which she was president. Well, too many experience. And Professor Judith also has been awarded Merit Award from the Society of Wetland Scientists. So, ladies and gentlemen, distinguished guests, let's watch Professor Judith Weiss' video presentation, please.
Hello, this is Judith Weiss. I'm talking to you from New York City. Sorry I can't be there, but we'll be talking about behavior changes that happen in contaminated environments and how these will have consequences for ecology. Contaminant effects can happen at many levels of organization.
Please wait, Prof. Judith, because we are having a technical problem with the video.
Do you want me to try to, I have it here.
Okay, so
The
would you like
audio
to share?
doesn't seem to be working.
Okay, please. So would you like to share from your side?
Well, if you could get this to work, that would be good.
Yeah,
but
yeah.
Otherwise,
Yeah, we are still trying here.
Should I try to put up my? Can I share my screen and find it? And let's see. I can't do this quickly. Perhaps you can.
Yes, oh, we are trying. We are trying now. Sudah. What's the problem?
I am trying to get back to see.
Behavior that can affect. Contaminant effects can happen at many levels of organization. For example, initial effects happen at the cell level, and then it translates up to the whole organism level, which may have effects at the population level, which can then have repercussions at the community level. What we're going to be talking about today is primarily at the organism level behavior, but behavior that can affect the population and the community, as we'll see. As behavior is a very sensitive effect at the organism level that can produce effects at higher levels of organization. First of all, I want to introduce you to our study sites. This is the state of New Jersey on the Atlantic coast of the U.S. This part of the state, the southern part of the state, is relatively undeveloped. It's not very urban and pretty clean. Whereas up here in the northern part of the state, there is a long history of a great deal of industry, manufacturing, heavy industry, that left lots of contaminants in the sediments, in the creeks and wetlands and estuaries up in the north. Our particular sites are Piles Creek and the Hackensack Meadowlands for polluted sites, Tuckerton as our reference or clean site. Just a look at what Piles Creek is like. Here's a sign that used to be there showing all these various chemical and petroleum manufacturing places. An aerial view shows all these petroleum storage things. In contrast, this is what it looks like at Tuckerton. It is a reserve, a refuge, and no heavy industry, and protected area down there. By background, the mummichug or killifish, I use the term interchangeably, scientific name Fundulus heteroclitus, from Piles Creek, we found do not grow as well and do not live as long as the same species in cleaner environments. And we thought that possibly behavior might play a role in causing the abnormal short life and reduced growth. So we set up experiments in which we would put 10 shrimp, grass shrimp are a major food for the killifish. We would put 10 grass shrimp in a tank with a rock for refuge. And then after the shrimp were acclimated, we'd put in 3 fish and wait a half hour and then come and record over time how many shrimp were left. So we're starting out with 10, and this is the Tuckerton predators here, and by the end of a half hour, they've generally consumed about 3 shrimp. In contrast, this is the Piles Creek fish that are only consuming on average about 1 shrimp in that time. And looking at their actual behavior, how hard they tried, how many strikes they made at the shrimp, we can see that the Piles Creek made fewer strikes. Watching them, you'd see they would try a bit and then give up, whereas the Tuckerton fish kept at it. They kept after the prey and were therefore able to catch more. behaviors we looked at was just general activity. And we can see here that the Piles Creek were only about half as active as the Tuckerton fish. So it's sort of the same idea as the poor prey capture. They're just slow. Looking at the idea that this might be caused by the environment, we did a switching experiment where we kept Piles Creek fish for 2 months in clean water, clean sediment, clean food, the Tuckerton environment. And we kept Tuckerton fish in a Piles Creek environment, aquarias with Piles Creek sediment, Piles Creek water, and grass shrimp from Piles Creek to eat. And after this time, we examined prey capture ability. And here we have this is the original Tuckerton shown in the previous graph. And these were found to have low brain mercury. Here are the others. These 3 lines are all basically the same. It includes the Piles Creek fish, the Piles Creek that lived in clean water, and the Tuckerton fish that lived in a Piles Creek environment. They're all comparable to each other. They all had high levels of mercury in their brain. So you can see the Tuckerton fish lost their good ability to capture prey. They're as bad as Piles Creek fish. And the Piles Creek that were living in the cleaner environment, which is these here, improved very little. I mean, there's not a significant difference here, but it's an indication that perhaps if they were kept in clean water longer, they might have improved much more. We did not do that, but I have a feeling that if we had kept them in clean water longer, they'd have improved. We also looked at predator avoidance, and this is now looking at the killifish as prey rather than as a predator. And in this case, we used blue crab, Calanectes sapidus, as the predator. The experiment put 10 fish in a tank with a rock to hide, and then we would later add 1 adult crab. And in this case, we would wait 2 weeks to look at what happened. And overall, Piles Creek fish in the 2 weeks, 23 were eaten, 7 were not eaten, while the Tuckerton, 12 were eaten and 19 were not eaten. So it's obvious here that the Piles Creek fish are easier for a predator to capture. So they're poor predators. They are easy targets for their own predators, and they're just slow in general activity. And we think that the impaired prey capture we observed could explain the poor growth that we had observed earlier, and also that the impaired predator avoidance could explain the reduced length of lifespan of the fish from Piles Creek. This is a field validation. We said, well, what are they really eating out there? Not just what are they eating and how fast are they eating shrimp in the lab, but what are they really eating there? And this is a pie chart of all the different things found in their stomachs. And this largest thing here for the Tuckerton fish is detritus. Detritus is decaying plant material, basically. And it's very easy to catch this. This doesn't run away. It doesn't take any effort whatsoever to go down and eat detritus. Here is the grass shrimp, comprising about 1/3 of the diet, and then other foods making up smaller amounts each. Here, on the other hand, is the Piles Creek diet. We see that detritus is now 3/4 of the diet. Most of the diet is this detritus, which has been shown to be not nutritious for killifish. Some animals do fine on a diet of detritus, but killifish don't do well on this diet. And here's the grass shrimp. Now it's down to almost only 10% of their diet, and then a few other things. So we see see, we found in the lab that they don't catch shrimp very well, and this is supporting that. They're not having much shrimp in their diet. Then we can ask, what about the shrimp? Are the shrimp also impaired in predator avoidance? So maybe if they're poor prey, they don't escape well, maybe things equal out. You've got the fish that are lousy predators and the shrimp that are easy to catch, and so things might equal out. That's a question. Maybe the Piles Creek fish eat less shrimp because there are less available. Maybe the population of shrimp at Piles Creek is small, and that explains why the fish don't eat so many. So we're now focused on the shrimp. And here is a look asking, are the shrimp at Piles Creek easier to catch than the shrimp at Tuckerton? And here's the answer is clearly no, they're equivalent. Actually, toward the end of the time, the Tuckerton fish begin to get caught more than the Tuckerton shrimp begin to get caught more, but there's no significant difference here. So there's no significant difference in the shrimp predator avoidance between Piles Creek and Tuckerton. Then asking the question, maybe there just aren't very many shrimp at Piles Creek. That's clearly not the case at all. We did a catch per unit effort, just trapping and counting animals, and overall catch per unit effort for Piles Creek mummy chugs was only about 1/3 of the Tuckerton mummy chugs. Killifish, same thing. The grass shrimp, on the other hand, were much more numerous at Piles Creek than Tuckerton. Furthermore, the size distribution of the shrimp at Piles Creek and Tuckerton showed that the Piles Creek shrimp were a whole lot larger than the Tuckerton shrimp. This would have been a big surprise if we didn't know all we know already about the killifish, which is the major predator in these tidal creeks. So the conclusions about the shrimp are that we have about 3 times more shrimp at Piles Creek, and they are larger. We have about 3 times more fish, killifish, at Tuckerton, and they are larger. And the low numbers and poor predatory ability of Piles Creek fish allows the shrimp to be more numerous and to grow larger, live longer, because they're not getting eaten so much. They are living a longer, happier life at Piles Creek. And therefore, the top-down refers to predator impact on their prey, outweighs any potential harmful effects that the pollution could have directly on the shrimp. Moving to another species, the blue crab. This is what was used as a predator in the studies looking at the killifish predator avoidance. These are fairly large predatory predominantly carnivorous crabs. And we've moved from Piles Creek, which was about here, to the Hackensack Meadowlands along the Hackensack River here. This is another highly polluted, but a much larger system in order to catch blue crabs and other species. This is also highly industrialized with heavy levels of metals and organic contaminants in the sediments. These signs are around in the meadowlands warning people against catching and eating crabs because they are contaminated with a variety of nasty chemicals. So we're comparing the prey capture ability of adult crabs from Tuckerton and the Hackensack Meadowlands. And we're also comparing the predator avoidance ability of juvenile blue crabs from both places. And in this case, we could use adult blue crabs as predators. They are cannibalistic species. So we don't have to find another species. We can do predator and prey as blue crabs using adults versus juveniles. First, looking at the adults, looking at how well they catch prey in the lab. And we can see the white is the Tuckerton and black is Hackensack. Comparable eating of rib mussels, comparable eating of fiddler crabs. But when it comes to prey that moves fast and is hard to catch, like killifish and blue crabs, there's a big difference. And you can see the Hackensack blue crabs are impaired in their ability to catch active prey. This is the stomach contents in the field. And you can see this again is supporting the evidence in the lab. A normal crab here has lots of crab parts, fish parts, and other things, living animals in their diet, whereas the Hackensack, the stomachs from Hackensack, is primarily sediment and detritus, which again, you just scoop up from the mud on the bottom, and plant material. So this is a highly unusual occurrence to see in a crab that is well known as a predatory crab. These crabs are impaired and not feeding on the things that blue crabs normally feed on. We did some transplants, comparable switching experiments, but we had two different ways of switching them. We had one where the crabs were in the lab receiving food and water from the other site. And then we had also actual transplants where we would put them in a cage at the other site and give them in the cage prey from that site. So here's looking at what happens to Hackensack. Bear in mind, this y-axis is on time to consume the prey. So the taller bar is a poor predator. The shorter the bar is, the quicker the animal is eating the prey. In this case, the predator is a big blue crab and the prey is little blue crabs. So here, this is the general baseline for Hackensack, taking a relatively long time. And whether you put them in cages in Tuckerton environment itself, or you put them in clean water and give them clean food in the lab, they get much better. Here's your Hackensack. The baseline for Hackensack is a very small bar, meaning it takes a relatively short time to eat the food. The ones that sat in Hackensack for the time got a lot worse, took a lot more time. The ones that were in the lab getting Hackensack mud, Hackensack food,
Tuckerton's supposed to be
worse, not quite as bad as the ones that were actually there. But again, a big difference from what a normal Tuckerton crab would do. Now, this is from the standpoint of the juvenile blue crabs. We're taking an adult predator and looking from the point of view of the juvenile. And here we have a y-axis that the higher is the number of survivors. And the number of survivors of Hackensack crabs is better of the juveniles than the Tuckerton. So here we have perhaps a surprise that the Hackensack juveniles did better than the adults. This is the opposite of what happened with the killifish when we looked at them as prey for blue crabs. So our conclusions, adults from Hackensack are poor at catching active prey. We switch the food or the habitat for 6 weeks, the behavior changes. Their stomach contents in Hackensack crabs are full of detritus and algae, highly abnormal. But the crabs are big and numerous. And it's possible that the fact that crabbing is not allowed and people are not catching them is partly responsible. The Hackensack juveniles, as we saw, the surprise, they're better at avoiding predation. And this fact may also partly support increased population growth if fewer juveniles are consumed, less cannibalism. The fourth and last species I want to talk about is the bluefish. Bluefish is a temporary resident. It's a commercial species like the blue crab, a commercial fishing species. This is a fish that lives most of its life in the ocean, and they spawn in the ocean, but then the little ones, the juveniles, move into estuaries for their early life, early juvenile stages, and they live in move in the spring. They spend the summer in the estuaries and then return back out to the ocean in the fall. So it's an interesting lifestyle. And the question we're asking is what happens to these juveniles when they spend their first summer in contaminated estuaries like the Hackensack Meadow? So we caught young of the year bluefish coming in from the ocean as they were entering the Tuckerton environment. So these are all fish collected from Tuckerton as the basis of the experiments I'm going to talk about. And we keep the fish in large tanks and fed them frozen fish. We fed them frozen Brevordia, which is the menhaden, and Fungilis, the killifish. We've been using killifish a lot in these experiments. And so they were getting the food either collected from Hackensack or from Tuckerton. So the Tuckerton ones were the controls. The ones that were fed Hackensack food, Hackensack frozen fish from Hackensack were the experimental ones. And at the end of the time, we quantified how much feeding they did. We looked at their swimming activity. We looked at the length and their length and weight. We had done the length and weight at the beginning to make sure all the experimentals and controls were the same size. Okay, so these are feeding experiments. This is not prey capture because they're eating frozen fish. So the prey has no, it's not prey, it's just food. And you can see here, this is seconds to capture a ration. Throw in X amount of frozen fish and time how long it takes to eat it all. And you can see here the first ration, the Hackensacks are taking a lot longer to capture. And again, the second ration and the third ration. These fish are very fast at eating the frozen food. So the Hackensack fed, these are not Hackensack fish, these are Tuckerton juvenile bluefish that are being fed food from Hackensack. Okay, and again, looking at how many prey fish they ate, the Tuckerton are capturing more food than the Hackensack. So they're not only capturing food faster, but they're overall catching more. And then we looked at their swimming. You can, in a tank like this with lines on it to count. And you can see again the swimming rate for the Tuckerton fed fish is much greater than for the Hackensack fed fish. And at the end of this time, we looked at the weight and the length, and we saw significant differences of the Hackensack having lower weight and lower length. Not a huge difference because they're being fed a ration of food. They're not having to catch their own food. So it's not an enormous difference, but it is statistically significant. There's no difference in condition index, which is the, ratio of length to weight. But they are significantly smaller after eating the different diets. Summing up the bluefish laboratory study, a reduction in feeding, eating less food, decreased spontaneous activity, reduced length and weight. And we think this reduced growth that they have and their reduced prey capture, reduced feeding, they will be less competitive when they go out to the ocean for the winter, where they will be mingling with bluefish that had spent their summer in a cleaner environment. We have some field data too, and I think this one is particularly interesting to me. Here's a juvenile bluefish. This is what a menhaden looks like. This is our killifish we started without at the beginning of the talk. We're looking at the contaminant level in the prey species. And these are the levels of the fish that we caught in the stomachs of the bluefish. So you catch the fish, kill it, you open up the stomach, and you have recognizable pieces of the fish that it ate before you caught it. So these are wild caught from the field. And we can measure what the contaminant levels are in the mummichogs and menhaden that had been eaten. And here are the levels. We could also capture with nets the mummichogs and the menhaden living in the Hackensack. And here's the levels for the fish that had not been eaten. So you can see here the ones that had been eaten have much higher levels of PCBs than the fish that were about twice as much, than the fish that were swimming around alive. This supports what we found before with lots of other species, that the contaminated fish are easier to catch. Here's a look from other field data, the bluefish sizes. We're now looking actually at Tuckerton fish and Hackensack fish. And you can see here a very significant, striking difference in size. Hackensack fish are smaller than the Tuckerton fish. They're weigh a whole lot less. They're 15% shorter and 44% weighing 44% less. That's a big difference. This is a much bigger difference than what we saw in the lab fish. The lab fish were somewhat smaller, but nowhere near this much smaller. But remember, these are fish from the field, the fish in the Hackensack that had to catch their own food, whereas the ones in the lab had frozen fish thrown into their tank. It's a whole lot easier than having to catch your own. Looking at percent bluefish with food in the gut, other people had studied bluefish, and overall they're seeing levels from the 60s to the 80s. And our Tuckerton bluefish had about 3/4 of them had prey in their stomachs when they were caught. In contrast, we see overall in the Hackensack, it's generally less than half, and down in the 20s as an average percentage of fish with food in their gut. So most of the fish that we caught out of Hackensack had empty stomachs, which is unusual. The nickname for the juvenile bluefish, they're called snappers because they're voracious predators. And obviously in Hackensack, they're not very voracious. So to summarize what we found, comparing killifish, bluefish, grass shrimp, and blue crab, feeding is down on all of them. I give some data from shrimp from Joshua Stout, who is not part of our study, but examined the same things with focusing on the grass shrimp. other aspects of their life. Field validation. Yes, killifish are full of detritus. Bluefish had empty stomachs. Blue crab was eating lots of detritus. So this is validating the fact that feeding is impaired. Predator avoidance was bad, in the killifish. We don't have data on the bluefish. The shrimp were equivalent, and the blue crab juveniles were actually better at predator avoidance. Activity was down for all the ones, this is from Stout's work, was down. We did not measure activity in blue crab. They spend most of the time sitting in the corner of a tank. We couldn't figure out how to do that. And in terms of size, the killifish and bluefish in the contaminated environments were smaller, noticeably smaller. The shrimp were larger. We attribute this to the fact that they are not getting eaten so much by the killifish. And the blue crabs are also bigger, and we attribute this to there being no fishing. That, again, their major predator, people, are not catching them so much. Population density, killifish was down. Bluefish, we have not got measurements. Shrimp were more common at Piles Creek, which we're attributing also to top-down, the fact that they're not getting eaten so much and they're living longer and growing to bigger size and having a larger population. Blue crab, we don't have data, but we suspect that the absence of fishing is allowing their population to be denser, but we don't know. One thing we didn't talk about at all in this study is reproductive effort, which we did see the killies in Piles Creek had reproduced sooner, so they had more reproductive efforts. Stout found that with the shrimp at Piles Creek. So that summarizes what we found. We have some thoughts about future work. We'd like to know, are organisms at higher trophic levels affected more than those lower down? Are certain taxonomic groups more successful? I mean, it appears from this limited number of species that crustaceans seem to do better than fish. Are certain traits associated with being more successful in contaminated areas? And then perhaps, are the particular contaminants at a given site more important in determining who is going to do better than whom? And I'm hoping other people will take up and look more at this. So thank you very much.
Okay, we just watched Professor Judith Wei's video presentations. After we would like to ask whether you want to give some comments on your video, Prof. Judith.
Yeah, there was one error that I tried to interrupt my talk, and the slide, the second, I don't know if you can go back to the second slide on the blue crab switch experiments. I think I said Tuckerton when I said Hackensack when I meant Tuckerton. What was on the slide was correct. It was when the Tuckerton crabs that were put in Hackensack became poor predators. I just had, misspoke, but the slide was correct. And I just wanted to clarify that. I interrupted myself when that slide was up. And if you were looking at reading what the slide said, that was correct. And if anybody wants to ask me more questions later, I'm happy to answer them. I know some people are putting some questions in the chat box. And it's, I'm happy to, talk with people afterwards by email if they want more information too. But we're going to have a discussion afterwards anyway. So
Yeah, that's great.
I'm managing to stay awake. It's after midnight here, and I'm trying to stay awake.
Okay, so we fixed the problem of the video. So before we go to the question and answer sessions, we still have one more speaker. The fourth speaker will be Young Evie Mevel. Yeah, please. I would like to read your profile or short biography, Dr. Young Evie Mevel. Young Evie Mevel, Dr. Young Evie Mevel is an aquaculture expert, aquaculture engineering and owner of Green and Blue, a post-organic aquaponic farm in Vientiane, Laos, PDR. Dr. Young Evie Mevel graduated with a PhD in aquaculture engineering and master degree in aquaculture and fish diseases at Auburn University, USA since 1979. He has been involved with most aspects of aquaculture in over 30 countries and 5 continents. In 1999, he spent a year as academic advisor in UNHAS for the Marine Science Education Project, providing assistance and advice to the institutions. And most of his work since has been in Southeast Asia and the Middle East, where he has developed large private aquaculture farms and has worked as national aquaculture expert at the Ministries of Agriculture and Fisheries in Bangladesh, Oman, and Cambodia. And Dr. Yang Evie Mevel also involved as president in the management of World Aquaculture Society, APC. Yeah. Dr. Yang Evie Mevel will present with the title IMTA, Remediations to Aquaculture, Environmental Impact. So please welcome to Dr. Yang Evie Mevel.
Yes, good morning.
Good morning.
I hope I will make sense because it's 6 in the morning here. I haven't slept all night looking at all your talks, and I have very small eyes, and I hope I will not be slurring too much. Anyway, so my name is Jean-Yves Mevel in French. Sorry, it's a difficult name to pronounce.
Sorry if misspelling.
But, there is no issue with that. I'm very thankful for the organizing committee to have invited me here, and I'm glad to meet again some old friends that I've not seen for a while. So I'm going to present a talk on IMTA, but first of all, I will share my slides. Wait a minute. What did I do? I had a scare because actually just a few minutes ago my connection died, but hopefully it will be okay now. And let's see if it's how does that work? Wait a minute. How do I share my? I'm supposed to click on share screen.
Can you?
And it doesn't.
Would you like us to share from our side, or would you like to share from your side?
I don't know why it doesn't share my screen.
Okay, we can help you from here.
That's a good start.
Please let me.
I just restarted my computer, so I'm not sure.
Yeah, we are trying to share from this.
I don't even hear you
here,
anymore.
Yeah.
Share screen.
Yes. Have you already sent me your presentation?
Yeah, but actually I modified it a little bit, so I would rather share it. But if we have to, we can look at the one I sent.
Wait, did you send me in the email? So I will try to find.
It's going to be too long to send it Zoom.
Oh, ini ada. Oh, wait.
My
I think Bu Sinta found.
I don't know why.
Bu Sinta found your presentation file, so we are trying to share from here.
Right. I think we'll have to go that way. It's not the final one, but it really doesn't want to do it.
I think it's probably as exhausted as you.
She's still downloading.
Melfest. Well, it's been up the whole night.
Yeah, and it should have worked. It refused. Okay, I think it's going to work now. It should work. Sorry for this. Do you see the presentation?
Yes.
All right. So I will start by presenting a little bit about the WS APC. In case some of you are not aware of that, we've been involved quite a bit actually with Indonesia, and we will hopefully do another meeting in Surabaya soon, but it was cancelled as it was planned for this September this year, but COVID-19 did its job. WOS was founded in 1969 and APC chapter in 1996. It's today the largest global professional organization for sustainable development of aquaculture, and it's promoting educational, scientific, and technological development for advancement of aquaculture through workshop and conference. There is two journals being published, Journal of World Aquaculture Society, which is really a peer-reviewed scientific papers, and the World Aquaculture magazine, which is more general publication on feature of what is going on in the news and so on. There is about 5,000 members all over the world, and everybody who is a member has access to all this, all what has been published through internet. So it's actually a pretty good platform, and I would recommend everybody to use it. And being the president, I'm president-elect, and next year I will be president. I would like to promote really interaction between the industry and the university, as now it's not such a good job at that. Hopefully we can get better cooperation between the industries and the university. So coming to my talk, aquaculture remediation through IMTA. This is a subject that has been pushed to me for the last few years. And basically, I don't want to spend too much time on this, but everybody knows that in agriculture or aquaculture, water, soil, and climates are going really the wrong way for us, and is deteriorating very, very fast. Water is disappearing, soil is disappearing, and climate don't make sense anymore. Actually, it's very cold right now. It should be warm here in France, so not so good right now. And the biggest buzzword we have lately is sustainability. We've heard that several times through several talks before. And basically, this is a study of how natural systems work and could remain working for as long as we can, or we manage them properly. And the ecological remain in balance. The ecology is remaining in balance through the future to be. In the 21st century, it really refers to a capacity for the biosphere and human civilization to coexist. And we are not doing such a great job. We are lying to ourselves in many ways and many examples. But certainly it's an issue that has been very dear to me lately and been involved in many aspects of it. Aquaculture, is it sustainable? Not really, actually. There is a lot of impacts. We've been seeing that throughout this conference. The first thing is that the aquaculture is generating a lot of waste and creating eutrophisation. Some good example is in ocean is cage farming. And this is getting pretty bad in the ocean because we are starting to have bigger and bigger dead zone. And that's a really big worry. And I've been reading some articles lately that are really scary. There is also an impact through poor management. Through poor maintenance of cage, you have escapes of fishes. You have a lot of increase in pathogenicity of organism around aquaculture. And the ocean are getting depleted anyway because we are still harvesting fish to produce fish through fish meal. So overall, aquaculture is not yet reducing so much the impact on fisheries. And we still depend a lot on byproducts of fisheries, but still it's impacting on the overall population of the fish. Something that is really bothering me a lot is that we are lying to ourselves. Offshore farming is one of the biggest thing right now, I would say, which we have this big new project for salmon. We are planning to move offshore. Sounds great. We don't see the pollution anymore. It's diluted, but we are still polluting the ocean. Everything, every waste is put in the ocean, and the ocean is doing the job for cleaning the mess. So we are hiding the pollution, but we are not avoiding the pollution. And this is not the proper way to go if we want to really improve our environment and reduce the impact of aquaculture on the environment in general. So in the last few years, the IMTA, Integrated Multi-Trophic Aquaculture, which I like to call agriculture also, it's a good way to optimize the aquaculture environment. What is it? Is that it seems logical and quite simple, but in fact to apply it is not so simple at all. Basically, we're using the waste of every trophic level to feed another trophic level, which in the same way will clean the environment and avoid accumulating waste of any one system. In this little diagram, you can see that we feed the fish, and all the waste from the fish, which is mainly fecal matter, but also ammonia in diluted form, we can feed different organisms. In aquaculture, I like, for instance, sea cucumber. We can have filter feeders, and for all the dissolved organic matter and so on, we can have algae, seaweed, and so on. So combining all these different trophic levels, we can actually produce much more and not eliminate, but reduce drastically the pollution and the impact on the marine environment. This is not a new concept. I like this picture, I don't know why, but basically a long time ago, the Aztec were doing multi-trophic aquaculture, agriculture in what they called, I forgot the name, guess I'm tired. But anyway, they were using all the resources from the aquatic environment and soil environment, and they were able to produce much more on a smaller volume, smaller surface area, and reduce the impact on the environment. Lately, in the last few years, we have a lot of brainstorming going on. We have some interesting design. I'm not sure they will ever be developed, but the idea of using every aspect of agriculture and aquaculture to recirculate all the waste which goes into the recycling and circular economy brings a lot of ideas, a lot of suggestions, a lot of projects that probably will never come through, but at least we're thinking about it, which is a good start. So The most well-known IMTA basically was to start with an aquaculture in mariculture in the sea, where you could grow fish. Then you could have what we call extractive aquaculture, like shellfish, which is going to filter organic matter, and then you can have seaweed, which will absorb the dissolved organic matter. We can see here an example in salmon industry, where you have on the left the salmon being produced in a fjord. Then on the right, you can see the seaweed hanging from some sort of raft, and and then probably some mussel in the back also to filter the environment. So instead of having one crop in one place, we have three crops, and the water quality is tremendously improved. This is getting to a point where in China you have huge extent of IMTA production. We can see that on hundreds of hectares in the sea, and more we are looking at seaweeds, and I enjoyed the talk before about that topic, where we need to work together in producing more with the waste of aquaculture. There is, beside the IMTA in mariculture, which people know quite well usually, I've been working with less known topic, but really interesting. I was advisor on the Sustainable Bioenergy Research Consortium, which regrouped Boeing and all kind of big company. And basically using brackish water aquaponic, we were able to produce actually biofuel that was used to have the first commercial flight in UAE 3-4 years ago. I forgot exactly when, but that was really a big deal at the time, and it was a commercial flight, which was the first one ever. So this project is still going on. I'm not sure what's going on now, but it's I think it's in Abu Dhabi, in UAE anyway, but it's in Abu Dhabi. It was a Masdar University, which now is Khalifa University, I think. And the facility was just ponds connected to the sea and some salt flats on which we were growing salicornia. The ponds were used to grow shrimp. The water was circulated through the shrimp pond, through the salicornia, and returned either to the pond and sometime to the sea, because there was a problem of hypersalinization. But the salicornia was then treated and transformed and produced biofuel. Here is I should have jumped the slides. Here is basically the system. You can see the interesting part is that the mangrove was not touched at all. We don't need to destroy the mangrove anymore for shrimp. That's known for a long time, luckily. But the water is basically mostly recycling and resent to the sea through the mangrove to avoid hypersalinification of the system. What I've been really involved mostly was really inland IMTA, which is called basically aquaponics. And when I arrived at the University of UAE University in Al Ain, I was really intrigued by already what they had done in aquaponic in the private or semi-private or agricultural school, but nothing was done in the university. And that was actually in Zayed Agricultural Center, which was a school for challenged people. And they were making some money, but that was not the purpose of it. But it was very pretty and quite attractive to look at. I found out that when this is too pretty like that, you are not making money, but that's another story. So what is aquaponic? Aquaponic is basically the production of fish and vegetables or other plants together. And and the water is recirculated through the fish and the plant and is treated by bacteria, nitrifying bacteria, Nitrosomas and nitrobacter, which will remove mostly, it remove nitrogen compound, ammonia, making a nitrite, which is a very good fertilizer for the plants. There is more subtleties into it with the possibility of mineralization of the waste, but to make it simple, that's how it works. Water gets dirtied by the fish, cleaned by the plants, and it keeps turning. So it's very economical. It saves in fertilizer, it saves in water, and the resource is really optimized. And we have 2 crops for the price of 1 almost. So I saw a lot of projects that really are beautiful and they make you dream for a very bright green future, right? The problem is the profit. The technology is there, we can do a nice job, but the profit has not always been there, unfortunately. And when you see some really nice production like that, you have to start thinking, well, these people are not selling the salad because that's too much salad at once. There is no staggering. That means it's all for show. And that's what I've seen a lot over the last few years. And that's what we need to change and start to think making money. In fact, I started my research project in UAU in 2012, and then I left in 2016 for different reasons and start my own business as well. But everything so far has mostly been based on the UVI aquaponics system, which works, but it might not be the best. And I'm not sure I'll have the time to talk about the technology there and the techniques, but there might be a better way to be more productive and than just a pure UVI system. But this is a basic concept to start properly. So the project was started in 2013, and we had actually 4 greenhouses like this one. And the trials look really promising, and the results actually were really good. We tried many different vegetables, but the salad, for instance, we were able to produce 1.2 tons of lettuce on 120 square meter of raft in 21 days. That's about 500 grams per square meter of raft per day of production of lettuce. So when you look at this number, you should be able to make money. But there is an extra cost that we don't always think. And selling 1.2 tons of lettuce is not always so easy. And it really depends of where you are. Tomato were working pretty well. For instance, we had 400 to 500 grams of tomato per square meter per day also on average. This is one plant, one tomato, huge, absolutely huge. The roots were unbelievable. And that's one day from one tree, basically. So this is working very well if we know what to do. The problem is actually it's not so simple. Everywhere you will see some rule of thumbs that looks very easy and all kind of numbers. I'm not going to give you these numbers, but and I'm lately trying to revamp a lot of equations, and there is nothing really well finalized in all the data we have. So it's not so simple to work, and it's therefore not so simple to make money, and that has been the biggest plague of the industry. In 2013, for instance, some people published a couple of things saying that the number of consultant and training course peddlers who claim to be able to show how you do were much more numerous than people who actually were successful in real commercial production. The other publication in 15 were saying that most purely aquaculture production was not making money. You had to do other activities around it to really survive. taken quite a while to start to see some production that were making money only on aquaponics. But the good thing about aquaponic, it's 2 times the understanding, 2 times the difficulty, but it's also 2 times more satisfying. You need to know plants, so you need to be an horticulturist. You need to know fish or whatever you grow, shrimp and so on. And you need to know microbiology because bacteria are going to be the system which keep um, the water in a good quality. So that's a lot of knowledge. That's one person very rarely has all together to start with. So anyway, I took the step in 2016 and I wanted to prove that we can make money on our own and build my own system in Laos. I found out that it's not so easy to make money with it still, especially in the time we are at right now. The production was pretty nice. It started quite well. I was able to break even, not being producing completely on all the system, but the quality was excellent. The taste was good. People liked it. So I was pretty excited about the future. The quality of the product is really, really nice. And there is no question that restaurants and people who want to take care of their health really look toward this kind of product. The quantity of plants we can grow is quite diverse, which is interesting. And we still don't know all what we can grow altogether. There is much more to be done and discovered. But unfortunately, with COVID-19, like many people, I had to close. I also personal issue to come back, death in the family and so on. So I came back to France. I'm stuck here since almost 1 year now. Cannot travel back. And so I had to close and put it on hold for now. But at least I know I'll be able to start again pretty soon if I can go back there. Meanwhile, I don't like to stay doing nothing, and I started to look around in France. And I've been really studying what can be done in the world and in Europe and so on. And basically, this kind of production is more than just food production. Of course, we can do food, fish, shrimp, and plants. But I'm looking at some new project now, and with a new Green Deal for Europe, for instance, for the European Union, governments are looking at much more than just production. First of all, with COVID-19, that's a tremendous boost for aquaponics because people don't want to depend on distant food. We had problem to get food from many places. We were short on food even in France. So now we are looking at developing local production. To give you an example, city in France have only a 1.8% autonomy in food production. Basically, anything happens in transportation, people go starving. So a lot of support from the EU and the government and the regions. And there is a complete aspect of social impact of this kind of project. And so I've started a project, for instance, in where I live here. And we are looking at all what can be around aquaponics. We have, first of all, a social function where we can have visit and restaurant around there, some tourism and eco sport around the place. Of course, we have the agricultural production. I won't talk too much about that. We have educational activities with school visits, extension work, and professional training and internship. We have all the environmental activities with producing green energy on the greenhouse. We have recycling the waste, and water is recycled and using water from the sky, from the rain. Of course, the economical functions are important for employment and support the local production. And R&D, we have already the support of the University of Lorraine develop what we call a campus food tech around this. So we have to look at the wider aspect of such an intricate production, and we can really bring the interests of the government. We are highly supported by the government, local region, and we hope to get the European Union involved pretty soon. And the other aspect is that we can do that in downtown. Basically, we are using an old 16th century citadel built under Louis XIV, if you know a little bit of history. And Verdun, where I am, is a world capital for peace. That was the place of a very huge battle in the First World War with 1 million people dead. You can see some cemetery there. And I call that an armistice with the environment. And we are planning to develop aquaponic here on the top of this fort, if you want. The city is all around here, so this is absolutely an urban project. And that's the beauty of an integrated production like that, is that you can address many aspects, and you don't have to do a huge production, but a more sophisticated and specialized production in urban environment. So I've been pretty fast, I think, but today what I can see is that we see the end of the tunnel, basically. We can start making money. We have the government supporting us in many, many cases. The technology has improved tremendously lately. In Europe, for instance, a lot of research has been done. I didn't know that before when I was in Asia. And we are looking at more aspects of mineralization of the waste, recycling even more the phases, where before we were only half doing that. But we still need to do a lot of research and development to achieve a fairly high level. I'm looking now at doing shrimp aquaponics inland with brackish water, and salicornia, for instance, or other halophytes. So we are looking at the beginning of an important industry, and we could, in long term, I think, avoid producing anything in the wild, and do everything in recycling system with absolutely zero pollution. And I think this is the future of the world if we want to survive much longer and be absolutely sustainable. I think I was a bit fast, but I hope there will be some questions. I thank you for your attention, and I return to Indonesia now for follow-up.
Thank you. Thank you very much, Dr. Yang, for the very nice presentation today. And now we finish all 4 speakers, and we move to the panel discussion sessions. And for the participant, please raise your hand in the chat, or you can give also questions to the chat room, and the speakers can directly answer your questions. Any questions? Let me check. Okay. Ladies and gentlemen, do you have any questions to... Hello? Test. Can you hear me, Yang?
Yes.
Sorry, maybe our voice was missed or disappear. Any questions, please? Yeah, from... Yeah, from Ibu... From Nepal, ya?
Oh yeah, because we just on the. Could someone please help me to read because there is something problem with my gadget. Yeah, there is one from Pak Husnul. Okay. One question from Pak Husnul in the in the room chat. Yeah. This for Professor Judith Weiss from Dr. Husnul Yakin from UNHAS. He's asking about how to apply animal behavior as a biomarker, such as swimming ability in the field study to determine the effects of pollutants. Professor Judith?
Yes.
Yes.
We looked into that, looking at predatory ability, and it wasn't always consistent. In highly polluted places, it was clearly impaired, but in moderately polluted places, it was very variable. So, at least with the species we looked at, I can't make a generalization, but with the species we looked at, it was quite clear in highly polluted places, but it was, it's very variable. And in places that were moderately polluted compared to places that were really quite clean, it was not clear. So in that way, I'm not sure, at least with the species we looked at and the behaviors that we looked at, it didn't seem to be that good for a biomarker, but I would not want to say other species.
Privately, so please open your chat. Okay, there is one raised hand from Dr. Indrajaya. Please, Pak Indrajaya. Oh, no, from Maros. Yeah.
Suaranya kecil, please make it bigger your voice.
Can you hear me?
No,
Huh?
we cannot hear you. It's not clear.
Hear me?
Yeah, that's good.
Okay. This not question, but I'd like to ask the opinion of from Ibu Nurjana. How are you, Ibu Dr. Nurjana? I would like to asking the opinion about how about using drone for imagery processing, particularly in the coastal area, land-based coastal area. Thank you, Bu.
You finish?
Yes, finish.
Okay. Bu Dr. Jana, do you have any comments? Please.
I think it is only.
We cannot hear your voice, Bu.
Okay, good night, everybody. I mean, good morning.
Good night,
good
Prof.
afternoon to you.
Judith. Have a nice
I'm
sleep.
gonna leave. So.
Good night. Thank you very much.
you all.
Thank you very much for joining us,
Okay,
and
bye.
thank you very much. Okay. Ibu Dr. Nurjana, do you finish?
Yeah. Indra Jaya, yeah, Mr. Indra Jaya, thank you, but I don't understand. I mean, you say something statement or any question? Which the question? I don't know.
Yes, yes. I mean, I would like to know your opinion about how using the drone, okay, for imagery processing, particularly in the area, land-based area in the coastal area.
Okay.
Thank you.
Okay, thank you. If you would like to know my opinion about the UAV, yes, the drone is very for the first reason is very need to know the specification of drone. We need, if you would like to use the imagery from a drone for research or for explore more specifically in a coastal area, we need to put or we need to use the multispectral camera. It means we need to put to add the accessory in the UAV, not only a UAV original, but we need to arrange and then put multispectral channel. It will be result the maximum classification of a coastal area. You see in my presentation, we can explore more specifically about the coastal habitat, where's the seagrass, where is the coral is better, of course, is better result because they have a high resolution. 4 centimeter is very high resolution. Yeah, that's all.
Thank you.
Can I say something about that? It's interesting because in where I've been working with aquaponic, for instance, and therefore agriculture, we use drone or fixed system on the roof of a greenhouse. And with the right spectrum, we can analyze the health of the plants. And therefore, we can determine which compound we should add, phosphorus or whatever. It's still in the development to some extent, but we are coming pretty far on this. And so these are technologies that will be applied in many, many aspects of agriculture and aquaculture for that matter, mangrove health or whatever you want. It's a matter of not the drone itself, it's on the matter of what camera you put on there, and then the computer system to analyze this green intensity or the different color we have.
Thank you.
It sounds like a good excuse, Pa Indra, for Rika Maros to purchase a fancy new drone, though.
Actually, I did that. I did that for my system, but unfortunately, I was near a telephone post or big post, and I lost my drone on the first minute I used it. So you have to think about it before you use your drone.
Yes.
Okay.
You need to take care when you operate this. Yes.
Expensive, expensive.
Okay, thank you.
Good time.
Ladies and gentlemen, the distinguished guests, do you have any questions? Please raise your hand. We still have more time to discuss. It's about 15 minutes. What time now in Brisbane, Prof. Nick? Nick, what time in Brisbane now?
It is very reasonable time. It's 3:10 in the afternoon.
Oh, okay.
Not like poor Jean over there, staying up all night.
It's getting early. I need to get up now.
Hey, since I'm talking at the moment, John, I love the fact that you could use the halophytes and salicornia, and I find them tasty. But I'm wondering, what about in Cambodia? Are they naturally, culturally consumed very regularly, or is it a high value?
No, I just bought some salicornia to eat. I never eat that. It's in my fridge right now. In Cambodia, I don't know what is the status now on there, but that was in the Middle East. It was in UAE and so on.
Okay.
There is a market to be developed. But I was just going to ask you because seaweed in aquaponic has to be thought carefully because there is a demand for shrimp and shrimp imported from the other side of the world is not a very good option actually, and the quality is not there most of the time. So we need to develop system. And in 1993 actually, yeah, before I went to UNHAS, I went to Israel, and that was the first time I saw integrated system with seaweed, mussels, and mahi-mahi as a recycling system. And basically it was sort of aquaponic if you want, but in salt water. So these are not new, but I don't know, we never really took off since. And now I think we need to look at, sea lettuce, for instance, is a good way to clean water. And we could have it in brackish water in center of, in Laos, which is a landlocked country, or anywhere. So we really need to look into all these things, and nobody is doing it right now, or not much.
In the Mekong, it's pretty challenging there with the freshwater and that gap between freshwater, so 2 parts per thousand up to 15. Even the best seaweeds don't, as in the most tolerant seaweeds, don't do well under 15 ppt, parts per thousand.
Yeah,
so, but that's why I like the salicornia, because in Australia they feed, they finish often the sheep, the lamb, on these salty bush and salicornia. So that's a very high value application if you have some livestock integrated as well.
Yeah, there is some good projects. So one of the project in France got $2.3 million, but just raising money for, and they just had a little pool to try and demonstrate their system. But this project have been done, like in Italy, they have grown Pacific white shrimp with sarcocornia, which is similar. I don't know that one actually. I know the name.
It's another. It's like salicornia. It's
it's very similar. Yeah, very similar, and it's working. So, but nobody is taking the step to go commercial. And that's the problem: is to go commercial to that, because that's something I wanted to say. Most people who start aquaponics, they either are horticulture, they want to produce salad, and they don't care about the fish. And I work with a partner who is really, he was working for the biggest salmon producer, and he's interested in the fish. He doesn't think we can make money with the plants. So, and as long as we cannot match each other and say we are going to make money with both aspects, and even more on the side, it's not going to work. I'm working on a project with the King of Morocco on OCP on again brackish water in the desert, and probably will be salicornia and some fish and shrimp. We'll see. So it's coming up. We'll hear about it very soon in odd place, because we can do that where there is very little water, and in place there nobody think about aquaponic or aquaculture at this time.
The fish farmer quickly becomes the vegetable farmer, because you only need about one fish for a ton of lettuce. So.
Yeah, not exactly, but yeah, it's true. But I was looking lately with somebody for 20 ton of fish and 20 ton of shrimp per day in aquaponic in Thailand. So we are going to see some, a lot of things are going to come up very soon. We just have to find some investors. Not a small job.
So all you need to do is go to your fridge now after the session and blanch very quickly, quick blanch, and then a fry, and then it's quite tasty. Salicornia.
Right. Yeah, exactly.
Just like asparagus. So.
Yeah. So I'll see how it does. I will do that tonight.
Very good. We've filled the time, Ibu Chitra,
Yes.
with talking about
Okay.
aquaponics. That's
so
good.
I think there is no more questions from the participants. And I have a question to Prof. Nick. Yeah, just want to know about your Asia program project in Indonesia. Due to the pandemic situation, so how you continue the project so you can make this project sustainable?
Yeah, it's just pure luck, really, Ibu. It was the final year of the project anyway, so our activities were winding down. There were some challenges, of course, with the team, and people had to operate in a COVID-safe way, and with the government, of course, that became clear, and some of the activities were modified. But for the most part, we were very lucky, because let's say 90% of the work that we had planned to do had already been done anyway. So everyone spent a lot of good time writing papers. So we have 30 publications from that project in international and Indonesian journals. So that is a great legacy for all the team. Yeah, I think that was part of the reason why we have so many publications, is because everyone stopped doing the physical activities. So maybe that was a benefit for us. I do worry though, and maybe you are asking this about going forward, and how we have project continuity, especially for the good things that came from the work in the next year or two. But hopefully everything will start opening back up more.
Okay, so very good. So you can produce 3 journals publications. Yeah, we hope from UNHAS, and maybe also the collaborator for this symposium from UMT, we can have a good collaboration between your university and from and also organized by World Aquaculture Society, focusing on aquaculture, and also collaboration with the Marine Coast Center for Marine Coast Small Islands, with headed by Dr. Nurjana. There is one participant raise hand. Okay, Johnny, Jody. Johnny D, Johnny, Johnny. JD, Pak JD, go ahead.
Yeah, thank you, Dr. Malina. So this is just a quick question for Dr. Mavel. One of the use cases that we've seen with our computer vision fish survey program is exactly to the implementation on the aquaculture environment, right? The use case that we are looking at is like the biomass measurement, which it can do now. We're also looking at the disease identification, like the lice on the fish's bodies, as well as in fact measuring the optimal feeding by identifying the residual food that is actually not used. So that is among the use cases that we think our solution can be applied in the aquaculture environment. Have you seen something similar to this used along your way?
Yeah, there is a lot of people working on that. There is a lot of system, more or less advances. There is tons of example, I would say. I think where you are going is maybe a little bit more advanced on the computer side, but on the practical side, on feed measurements. Interestingly enough, for instance, we can measure the sound of the noise of the shrimp eating in a pond and analyzing how much they eat, how many shrimp there is. A lot of information just from the sound that they are making, tang, 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 whatever they make. And that's something I remember talking about that 30 years ago, and there was no technology for that, but we could hear it. But the computer where we are still with Apple IIc, So now you can see, and now there is a technology that is a couple years old, maybe more, that really use this to define how much we need to feed the shrimp. Counting the fish, evaluating the dimension, the size of the fish. They can go through a circle receptor, whatever. It measures the density and everything. A lot of the information are there. The problem is not the technology. We have a lot of technology that needs to be applied. It's just a matter of talking between practical people on the field like me and you, for instance. We can define what we need. The problem is the cost. We cannot sell the fish more than what we sell it already now. Because if we don't sell the fish, we are going to give the chicken producer are going to sell their product. And that's the biggest competition with fisheries and aquaculture is of the chicken. And so we cannot raise the fish definitively, the price of the fish definitively. So we have to find a way to get to a decent price to install all these things. Now, I've seen most of the very high-tech farm closing. They can't make the money. We forget that. And everywhere I've seen it, and that's where also there is I'd like to talk about that when we have in a meeting with university, is that everybody who does research look at maximum productivity is not necessarily what makes money. And many people forget that we need to make money to survive at anything we do, research included. So the technology is there, and with AI and so on, we are improving, going very quick. Now, can we make it economically? That's the problem. But yeah, there is tons of application in water quality, management of water flows, and management of density of plants, and all kind of things. I have tons of ideas. I don't have the money to pay you to do that. That's very simple.
That's very true, exactly. I mean, we have been trying to look for investor to basically to improve, to reach our vision with our software. That has always been the challenge. That's why the initial collaboration, we were hoping like the government, the Australian Institute of Marine and Science, that's usually, they're probably the one with the deep pocket, right? So we are hoping that's that type of collaboration that we can get together.
Yeah, and like I said before, talking about WS, we need to develop this interaction between industry and research and the government. There is nobody looks next door, and that's a pity. And nobody also looks at the technology from another field that can be applied. Everybody is really narrow-minded in their field, and we are lacking a lot of things. And I've been trying to keep an horizontal mind where I can bring electronics and mechanic and all that in one place. But very few people are doing that, and it's quite difficult because depending on where you are, you don't have the same resources. So you have to be very adaptable, and eventually we'll get there, but there is still a lot of work to be done. The communication is what is lacking mostly in our field, maybe in other field, I don't know.
Okay.
Thank you.
Yeah,
Dr.
thank you
Melville.
very much. We still have one last questions because the time running so fast, so we will end this the second sessions from Latif Sahubawa, Department of Fisheries, University of Gajah Mada. The question is for Dr. Jan. How is the application of aquaponics technology to aquaculture business for sustainable agriculture activities in Thailand compared to other countries, including Indonesia?
In Thailand, it's funny. There is quite a few things already, but it's relatively small. Aquaponic in most places in the world is still very artisanal, I would say, or amateurish. There is very few big production yet. China has a very big production of 120 hectares. It's just, but that's China. Everywhere in the world is pretty small. Indonesia has quite a few things actually, but also kind of amateur people doing a good job, I think, from what I see. But we need to go to the next step of being more industrial. And right now, for instance, I've been working in France for the last few months. I think the minimum size would be about 4,000-5,000 square meter, half an hectare of plant for to the fish. And basically, I estimate we could produce about 20 tons of fish for about 4,000 of square meter of greenhouse, knowing that there is only 40% of plant surface area for the total greenhouse surface area. Greenhouse technology, that's another aspect, and it's coming through fisheries, it's coming through aquaculture. I mean, you can see that above shrimp. It's definitively about aquaponics, but that's a technology that most aquaculturists don't know anything about, and I don't know much about it. I'm learning because I have to. But there is still a lot to learn, and there is no country where you have specialists that know everything for that topic. And that's the biggest problem everywhere.
Okay, thank you very much.
Thank you very much, Dr.
Thank you very
Jin.
much. Yeah. Now we are in the end of the second sessions. Before we end, I would like to summarize some topics from the first session and the second sessions. From the 8 speakers, we see that there are focusing on two major things. That the first one is innovations, and the second one related to environment. And as we know that the COVID-19 pandemic has affected the world economy, and Indonesia has not escaped its impacts. The marine and fisheries sector has suffered significantly in Indonesia, the largest archipelagic country in the world, where the marine and fisheries sector plays an important role in contributing to food security, livelihoods, and foreign exchange earnings. So that's why the Faculty of Marine and Fisheries Science, in collaborating with University Malaysia Terengganu, now is organizing one international 8th international and 4th international symposium with the theme acceleration of management of marine and fishery resources in achieving sustainable development goals in the era of the COVID-19 pandemic. The two measuring is innovations and environment. From the Professor James Bell, he focused on how to innovate state-of-the-art oceanographic instruments for marine observation, in-depth analysis, or in this case is DNA analysis of marine biota integrated with a marine geography information system. So an integrated and multidisciplinary approach is there is a must on this innovation. And related to the innovation, we also took some lessons from Johnny De Ong and from Satoshi and Professor Nicholas Powell. That from Johnny De Ong, he showed that machine learning and artificial intelligence is very important for marine science and fisheries sustainability. And for the Dr. Satoshi, he used GC to reduce the H2 hydrogen sulfide in the topic of remediation of coastal marine sediments using granulated coal ash. And Professor Nicholas Powell also showed that there are opportunities for Indonesia to enhance economic, environmental, and social well-being by strengthening innovations on seaweed. This is in order to achieve sustainable development goals. And related to innovation, we show Dr. Nurjana also showed her research result on spectral signature and using spectral signature and map coastal habitat on small island Spermonde archipelago by satellite remote sensing. And the last related to the innovation for the marine and fisheries sector is from Dr. Yang Evie Mevel. He showed that the most promising option nowadays to do aquaculture with minimum human impact on his environment is integrated multi-tropic aquaculture practice, or IMTA. And the second major focused on environment, we could found some lesson from Muhammad Faisal Hizam. He showed how to analyze or minimize some potential risk, the impact of bycatch and to the fish and its potential risk. Yeah. And the second is Professor Judith Weiss. Professor Judith Weiss also studied for common estuarine animals, comparing the predator or prey behavior of population living in highly contaminant versus clean environment in New Jersey. And she found that looking at the levels of mercury and PCB in the killifish and Manhattan prey from the stomach, reflecting that the earlier observation that more contaminated fish are easier to capture. So impairment in prey capture or predator avoidance behavior were thus found to have consequence at the population and community level. And the last also from Asoka, Dr. Asoka. Yeah, I have mentioned before, this is we can put it into innovation and environmental focus. Thank you very much. We would like to thank to the all invited speaker for from the first sessions and the second session to the 8 invited speaker. A warm thank you to you, and thank you very much for joining us today and give some very fruitful discussion and very important information and insight knowledge to us. And a warm thank you to all academics, researchers, and observers of the marine and fisheries sector for their participation in the 2020 Hasanuddin University Marine and Fisheries Symposium, and all the parties in collaboration with University Malaysia Terengganu, whose support and cooperation contribute to the success of this symposium. We thank you to all, and we also thank to the committee that makes this event happen. We say sorry if there are some mistakes from us as a committee to all invited speakers and to all participants today. And we hope that all the contribution made will be a benefit and promote the development of the marine and fisheries sector towards the achievement of the sustainable development goals, especially sustainable development goals 14. Thank you very much once again for all the invited speakers. We are very happy to have you today, and looking forward to any collaboration with you all from UNHAS and from UMT and for the all institutions from university, research institute, and from government or private sector. We are looking forward to research collaboration later on. Maybe this year we can try to overcome some problems related to the pandemic era situation in our country. Thank you. By saying Alhamdulillah, I close this second session. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh. And I give it the session next to Master of Ceremony Suci and Adnan.
Thank you, Ibu Tito.
Thank you, Pak Prof. Nick.
Thank you.
Thank you semua. Thank you semua.
Halo, Dr. James. Yes.
Thank you, Ms. Asmi Chitra Malina, SPE Master of Agriculture, PhD, for having incredibly lead this session.
Ladies and gentlemen, before we begin the party session, we kindly remind all of the participants that the party session will be held at 2 p.m., so the participants can join the room at 1:40 p.m. Furthermore, presenters and non-presenters who have registered as participants in this symposium will be given an e-certificate by the committee, which will be sent via email. Another information you can see in the display after closing the plenary session.
Ladies and gentlemen, we are now near the closing of this event. We sincerely thank you for your kind attendance and participation during the session. We hope to see you again in our 5th International Symposium on Marine and Fisheries next year. Hopefully, this event will be significantly beneficial for all of us. As Master of Ceremony, we represent the committee, apologize for any mistake during this event. Wassalamualaikum warahmatullahi wabarakatuh.
Warahmatullahi wabarakatuh.